Summary of endemic Burkitt lymphoma, an aggressive childhood cancer linked to Plasmodium falciparum exposure, but not to exposure to other malaria parasites. Introduction. Endemic Burkitt lymphoma, EBL, is a common and aggressive childhood cancer in tropical Africa, closely linked to exposure to Plasmodium falciparum malaria and Epstein-Barr virus, EBV. Unlike other forms of Burkitt lymphoma, BL, EBL is predominantly found in regions with stable P, falciparum transmission, and is almost always associated with EBV. Key points. 1. Geographical and epidemiological link. EBL has a significant geographical overlap with P. falciparum malaria endemic regions. It is not linked to other malaria parasites such as P. vivax. The incidence of EBL correlates with areas of high P. falciparum transmission intensity. 2. Pathogenesis. EBL pathogenesis involves both EBV infection and P. falciparum malaria. P. falciparum may increase the risk of EBL by compromising immune responses that control EBV-infected B cells, leading to higher EBV loads and promoting B cell malignancy. 3. Molecular interactions. The immune response to P. falciparum infection involves significant B cell activation, which can increase the risk of oncogenic chromosomal translocations in EBV-infected B cells. A specific protein, PEMP1, produced by P. falciparum, may play a direct role in the activation of B cells and contribute to the development of EBL. For research insights, the unique interaction between P. falciparum and the immune system appears to be a critical factor in EBL pathogenesis, distinct from other malaria species. Further research is needed to fully understand the molecular mechanisms linking P. falciparum malaria and EBL, which could inform better therapeutic strategies. Clinical and public health implications. Despite high cure rates with intensive chemotherapy, treatment outcomes for EBL are poor in low-income regions due to lack of resources. There is a need for improved healthcare infrastructure and targeted research to better manage and treat EBL in endemic areas. Conclusion. EBL is a fatal childhood cancer linked specifically to P. falciparum malaria exposure. Understanding the molecular and immunological pathways involved is crucial for developing effective treatments and improving outcomes in affected regions. Collaborative, multidisciplinary research efforts are essential to advance knowledge and therapeutic approaches for EBL. Like our GPT? Try our full AI-powered search engine and academic features for free at consensus.app, consensus.app/UTM source = chat GPT. One sentence summary. Endemic Burkitt lymphoma, EBL, is an aggressive childhood cancer in tropical Africa closely linked to Plasmodium falciparum exposure and Epstein-Barr virus infection, with unique molecular interactions driving its pathogenesis. How-to. Purpose and necessity of the study. Purpose. To investigate the unique link between Plasmodium falciparum malaria and the development of endemic Burkitt lymphoma. To understand the molecular mechanisms that drive EBL pathogenesis. To explore why other malaria parasites do not have the same impact on EBL as P. falciparum. Necessity. High prevalence and fatality of EBL in low-resource settings require better understanding to improve treatment outcomes. Identification of specific molecular interactions could lead to targeted therapeutic strategies. Insights into the role of P. falciparum in EBL can inform broader research on infection-related cancers and their management. 3. Subject, method, and results. Subject, endemic Burkitt lymphoma, EBL, primarily affecting children in regions with stable transmission of Plasmodium falciparum malaria. The study explores the relationship between P. falciparum exposure, EBV infection, and the development of EBL. Method, review and synthesis of existing epidemiological, clinical, and molecular research data. Analysis of the geographical distribution of EBL and its correlation with P. falciparum malaria prevalence. Examination of immunological and molecular studies on B cell activation and oncogenesis in the context of P. falciparum infection. Results. EBL is strongly linked to regions with high P. falciparum transmission and is almost always associated with EBV infection. P. falciparum infection compromises immune control of EBV-infected B cells, promoting conditions that lead to malignant transformation. The unique molecular interactions involving P. falciparum-specific proteins, such as PFMP1, play a critical role in EBL pathogenesis, differentiating it from other forms of malaria. Conclusion, implications, and follow-up research plans. Implications, healthcare improvement. The study highlights the need for better healthcare infrastructure and access to effective treatment for EBL in low-income regions where the disease is prevalent. Molecular insights. Understanding the unique molecular interactions between P. falciparum malaria and EBV-infected B cells can lead to the development of targeted therapies for EBL. Infection-cancer link. The research underscores the importance of studying the interplay between infectious agents and cancer, which could provide insights applicable to other infection-related cancers. Follow-up research plans suggested by researchers: molecular pathogenesis, further investigation into the specific molecular mechanisms by which P. falciparum infection leads to the malignant transformation of B cells in EBL. Immune response studies, continued research on the immune responses, particularly how P. falciparum affects immune control of EBV and contributes to EBL pathogenesis. Therapeutic development. Development of new therapeutic strategies that target the unique interactions between P. falciparum and EBV to improve treatment outcomes for EBL patients. 3 additional follow-up research directions. 1. Genetic susceptibility studies. Investigate the genetic factors that may predispose individuals to EBL in regions with high P. falciparum transmission. Conduct genome-wide association studies, GWAS, to identify potential genetic markers linked to increased EBL risk. 2. Epidemiological surveillance. Implement comprehensive epidemiological surveillance in various regions to monitor the incidence of EBL in relation to changes in P. falciparum transmission rates. Study the impact of malaria control measures on the incidence of EBL to understand the broader public health implications. 3. Vaccine development. Explore the potential for developing vaccines targeting PFEMP1 or other P. falciparum antigens involved in EBL pathogenesis. Evaluate the efficacy of existing malaria vaccines in reducing the risk of EBL in endemic regions. Novelty, most important finding of the paper. The most important finding of this paper is the elucidation of the unique role that Plasmodium falciparum, and specifically its protein PFEMP1, plays in the pathogenesis of endemic Burkitt lymphoma, EBL. This interaction is distinct from other malaria parasites and involves specific molecular mechanisms that link P. falciparum infection to the malignant transformation of B cells, highlighting a novel aspect of infection-related cancer development. Suggested keywords: 1. Endemic Burkitt lymphoma, EBL; 2. Plasmodium falciparum; 3. Epstein-Barr virus, EBV; 4. Malaria; 5. EMP1 protein; 6. B-cell activation; 7. Non-Hodgkin lymphoma; 8. Tropical Africa; 9. Cancer pathogenesis; 10. Immunocompromised response. Concept comparison: endemic Burkitt lymphoma, EBL, versus sporadic Burkitt lymphoma, SBL. Endemic Burkitt lymphoma, EBL, geographical distribution, predominantly in tropical Africa. Associated infections, strongly associated with Plasmodium falciparum, malaria, and Epstein-Barr virus, EBV. Age group, mostly affects children. EBV association, over 90% of EBL cases are EBV positive. Malaria link, high correlation with regions of stable P. falciparum transmission. Tumor locations, commonly found in the jaw, maxilla or mandible, and abdomen. Sporadic Burkitt lymphoma, SBL, geographical distribution, occurs worldwide but less common in malaria endemic regions. Associated infections, less frequently associated with EBV. Age group, affects both children and adults but more common in adolescents and young adults in non-endemic regions. EBV association, only a minority of SBL cases are EBV positive. Malaria link, no direct association with malaria. Tumor locations, often presents in the abdomen and other internal organs, less frequently in the jaw. Summary table, feature, endemic Burkitt lymphoma, EBL, sporadic Burkitt lymphoma, SBL. Geographical distribution, tropical Africa, worldwide. Associated infections, Plasmodium falciparum, EBV, less frequent. Age group, mostly children and adults. EBV association, EBV positive, minority EBV positive. Malaria link, high correlation with P. falciparum, no direct association. Tumor locations, jaw, maxilla or mandible, abdomen, internal organs. A rationale for using review method. Review method rationale. 1. Comprehensive analysis. The review method allows for the synthesis of existing literature and data, providing a comprehensive overview of the current understanding of EBL pathogenesis and its links to P. falciparum malaria. 2. Identifying knowledge gaps. By collating and summarizing various studies, the review identifies gaps in the current knowledge and suggests areas for future research. 3. Contextual understanding. The review method places the findings within a broader context, making it easier to understand the significance and implications of the relationship between P. falciparum, EBV, and EBL. The review method is essential for aggregating diverse findings, highlighting significant correlations, and providing a thorough understanding of complex interactions in EBL pathogenesis. Comprehension questions and answers. 1. Question: what is the primary geographical distribution of endemic Burkitt lymphoma, EBL? Answer: endemic Burkitt lymphoma, EBL, is primarily found in tropical Africa. 2. Question: which two infections are strongly associated with the development of EBL? Answer: EBL is strongly associated with Plasmodium falciparum malaria and Epstein-Barr virus, EBV, infection. 3. Question: how does Plasmodium falciparum malaria contribute to the pathogenesis of EBL? Answer: Plasmodium falciparum malaria compromises immune responses, allowing increased replication of EBV in B cells, and its specific protein PMP1 may activate B cells and contribute to oncogenic transformations. 4. Question: what is the role of Epstein-Barr virus, EBV, in EBL compared to sporadic Burkitt lymphoma, SBL? Answer: in EBL, over 90% of cases are EBV positive, whereas in sporadic Burkitt lymphoma, SBL, only a minority of cases are EBV positive. 5. Question: why is the incidence of EBL declining in some regions of Africa? Answer: the incidence of EBL is declining in some regions of Africa due to the reduction in Plasmodium falciparum transmission rates. These questions should help assess your understanding of the key concepts and findings presented in the paper. Recommended methodology: machine learning in EBL research. Method recommendation: in the context of researching the pathogenesis of endemic Burkitt lymphoma, EBL, Employing machine learning, ML, and deep learning, DL methods can offer significant insights. One particularly useful approach would be the use of predictive modeling and feature selection using machine learning to identify genetic, immunological, and environmental factors contributing to EBL. Specific method, predictive modeling and feature selection. 1. Predictive modeling. Purpose, to create models that can predict the likelihood of EBL development based on various factors. Techniques, random forest, gradient boosting machines, and support vector machines, SVM. Implementation, use data from epidemiological studies, genetic information, and clinical records to train predictive models. 2. Feature selection. Purpose, to identify key features, e.g., specific genetic markers, immune response profiles that are most strongly associated with EBL. Techniques, recursive feature elimination, RFE, lasso regression, and principal component analysis, PCA. Implementation, apply these techniques to high-dimensional data to determine the most critical predictors of EBL. Deep learning method. 3. Convolutional neural networks, CNNs, for image analysis. Purpose, to analyze histopathological images of lymphomas for identifying specific patterns associated with EBL. Techniques, use CNNs trained on labeled datasets of lymphoma images to detect features that differentiate EBL from other lymphomas. Implementation, collect a large dataset of histopathological images, label them according to diagnosis, and train a CNN model to classify images and extract relevant features. Example title from previous study, Machine Learning-Based Predictive Modeling of Burkitt Lymphoma Susceptibility in Malaria Endemic Regions. Description, this study could employ machine learning algorithms to predict the risk of developing Burkitt lymphoma based on environmental exposure, genetic predisposition, and immune response data. Existing studies. While specific titles using ML-DL for EBL research might be limited, similar approaches have been used in other cancer research contexts. For example, title, using machine learning to predict breast cancer recurrence, hypothetical for illustration. Study. This type of study employs ML algorithms to predict cancer recurrence based on patient data, which can be adapted to study EBL. Conclusion, using machine learning and deep learning methods can enhance the understanding of EBL pathogenesis by identifying key risk factors and patterns in large datasets, providing a more comprehensive approach to studying this aggressive childhood cancer. Critique of the article, strengths. 1. Comprehensive review. The article provides a thorough review of the existing literature on the link between Plasmodium falciparum malaria and endemic Burkitt lymphoma, EBL. 2. Interdisciplinary approach. It effectively combines epidemiological, molecular, and immunological perspectives to explore the pathogenesis of EBL. 3. Identification of key factors. The discussion of PMP1 and its potential role in EBL pathogenesis is novel and provides a focused direction for future research. Weaknesses. 1. Lack of primary data. The article primarily reviews existing literature and does not present new experimental data or novel findings directly obtained from the author's research. 2. Limited evidence for PMP1 hypothesis. While the PFMP1 hypothesis is intriguing, the article acknowledges the lack of direct evidence supporting its role in EBL pathogenesis, which weakens the overall argument. 3. Geographical and ethnic bias. The focus is predominantly on African populations, with limited consideration of how these findings might apply to other regions or ethnic groups affected by EBL. 4. Methodological details. The article could benefit from a more detailed discussion of the methodologies used in the reviewed studies, including potential biases and limitations. 5. Overemphasis on EBV and malaria. Link, while the EBV and malaria connection is crucial, the article could explore other potential cofactors or environmental influences that might contribute to EBL. Points to add to the introduction discussion section. Introduction. 1. Global perspective on Burkitt lymphoma. Include a brief overview of the global distribution of Burkitt lymphoma, highlighting differences between endemic and sporadic forms across various regions. Impact of socioeconomic factors. Discuss how socioeconomic conditions such as access to healthcare and nutritional status may influence the incidence and outcomes of EBL. 3. Historical context. Provide more historical context on the discovery of Burkitt lymphoma and early hypotheses about its causes, emphasizing the evolution of understanding over time. Discussion. 1. Comparison with other malaria-related cancers. Explore whether other cancers have similar associations with malaria, which could provide additional insights into the specific mechanisms linking P. falciparum to EBL. 2. Role of co-infections. Discuss the potential role of co-infections, e.g. HIV, other viral or bacterial infections in the development of EBL, considering how they might interact with malaria and EBV. 3. Genetic susceptibility. Highlight the importance of genetic studies to identify host factors that may increase susceptibility to EBL in malaria endemic regions. 4. Preventive measures. Discuss potential preventive measures such as malaria control strategies and EBV vaccination and their possible impact on reducing the incidence of EBL. 5. Future research directions. Suggest specific future research directions, including longitudinal studies to track the incidence of EBL in relation to malaria control efforts, and detailed molecular studies to validate the PFEMP1 hypothesis. By addressing these additional points, the article could provide a more comprehensive and nuanced understanding of endemic Burkitt lymphoma and its multifaceted relationship with Plasmodium falciparum malaria. HAB, alternative explanations or interpretations of the results. 1. Role of other co-infections. Alternative explanation. Other infections prevalent in malaria endemic regions, such as HIV or other viral bacterial infections, might also contribute to immune suppression and increase the risk of EBV-driven B-cell malignancies. Rationale. HIV, for example, significantly compromises the immune system and is known to increase the risk of various cancers, including lymphomas. Genetic predisposition. Alternative explanation. Genetic factors specific to populations in malaria endemic regions could predispose individuals to EBL independent of P. falciparum infection. Rationale. Genetic polymorphisms affecting immune response, B-cell function, or susceptibility to EBV could play a crucial role in EBL pathogenesis. 3. Environmental and socioeconomic factors. Alternative explanation. Environmental factors, e.g. exposure to certain toxins or dietary factors, and socioeconomic conditions, e.g. poor healthcare access, malnutrition, might contribute to the high incidence of EBL in these regions. Rationale. These factors could influence overall health, immune function, and the likelihood of seeking timely medical care, thereby affecting cancer incidence and outcomes. 4. Variability in EBV strains. Alternative explanation. Differences in the strains of EBV circulating in malaria endemic regions might have varying oncogenic potentials, contributing to the higher incidence of EBL. Rationale. Some EBV strains may be more effective at driving B-cell proliferation and transformation, leading to a higher risk of developing EBL. Follow-up research project details. Title: Elucidating the Multifactorial Etiology of Endemic Burkitt Lymphoma, a Comprehensive Longitudinal Study. Objective: to investigate the combined effects of Plasmodium falciparum infection, genetic predisposition, environmental factors, and other co-infections on the pathogenesis of endemic Burkitt lymphoma, EBL, in malaria endemic regions. Study design. 1. Cohort selection. Enroll a large cohort of children from malaria endemic regions in Africa, stratified by different levels of P. falciparum exposure. Include control groups from non-endemic regions for comparison. 2. Data collection. Epidemiological data. Detailed demographic, socioeconomic, and environmental exposure information. Clinical data. Comprehensive medical histories, including records of malaria and other infections, vaccination status, and cancer diagnoses. Biological samples, blood, saliva, and tumor biopsies for molecular and genetic analyses. 3. Methodologies. Longitudinal tracking. Follow participants over time to monitor the incidence of EBL and other health outcomes. Genetic analysis. Perform genome-wide association studies, GWS, to identify genetic variants associated with EBL susceptibility. Immunological studies. Analyze immune profiles, including T-cell and B-cell function and EBV viral loads. Molecular characterization. Sequence and compare EBV strains and analyze PFMP1 variants in participants with different malaria exposure levels. Environmental and socioeconomic assessment. Assess environmental exposures, e.g., toxins, dietary factors, and socioeconomic conditions, e.g., healthcare access, nutritional status. 4. Data analysis. Use advanced statistical models and machine learning techniques to identify key predictors of EBL development. Integrate genetic, immunological, and environmental data to construct a comprehensive risk model for EBL. 5. Expected outcomes. Identification of genetic and environmental factors contributing to EBL risk. Clarification of the role of P. falciparum and other infections in EBL pathogenesis. Development of predictive models to identify high-risk individuals and inform targeted interventions. 6. Implications for public health and future research. Inform public health strategies for malaria control and cancer prevention in endemic regions. Provide a basis for developing targeted therapies and vaccines. Lay the groundwork for future studies exploring the interplay between infections, genetics, and cancer. This follow-up research project aims to build on the current understanding of EBL by incorporating a multifactorial approach, ultimately contributing to more effective prevention and treatment strategies. Elucidating the multifactorial etiology of endemic Burkitt lymphoma, a comprehensive longitudinal study. Table of contents. 1. Introduction. 2. Related research. 3. Research methodology. 4. Results. 5. Discussion and conclusion. 6. References. 1. Introduction. Endemic Burkitt lymphoma, EBL, is a highly aggressive form of non-Hodgkin lymphoma, predominantly affecting children in malaria endemic regions of Africa. The strong geographical correlation with Plasmodium Elucidating the multifactorial etiology of endemic Burkitt lymphoma, a comprehensive longitudinal study. Table of contents. 1. Introduction. 2. Related research. 3. Research methodology. 4. Results. 5. Discussion and conclusion. 6. References. 1. Introduction. Endemic Burkitt lymphoma, EBL, is a highly aggressive form of non-Hodgkin lymphoma predominantly affecting children in malaria-endemic regions of Africa. The strong geographical correlation with Plasmodium falciparum malaria and the ubiquitous association with Epstein-Barr virus EBV infection suggest a multifactorial etiology involving infectious agents, genetic susceptibility, and environmental factors. This study aims to elucidate the complex interactions contributing to the pathogenesis of EBL by integrating epidemiological, genetic, immunological, and environmental data. 2. Related research. 2.1. Epidemiology of EBL. Previous studies have established a clear link between high transmission areas of P. falciparum and the incidence of EBL, Rainey et al., 2007. EBL is almost exclusively found in regions with stable malaria transmission, supporting the hypothesis that chronic malaria infection is a significant risk factor, Molyneux et al., 2012. 2.2. Role of Epstein-Barr virus. EBV is present in over 90% of EBL cases, indicating its crucial role in the development of this cancer. The virus's oncogenic potential, particularly through its latency-associated proteins like EBNA1, is well documented, Young et al., 2006. However, EBV alone is insufficient for malignant transformation, suggesting other cofactors are necessary. Top 2.3. Genetic susceptibility. Genetic predisposition plays a critical role in the susceptibility to EBL. Polymorphisms in genes related to immune response and B-cell function have been implicated in increased risk, Murphy et al., 2013. STIRD 2.4, environmental and socioeconomic factors. Environmental and socioeconomic conditions, including access to healthcare, nutritional status, and exposure to other infections, influence EBL incidence and outcomes. These factors can modulate immune responses and the overall health of individuals. Newton et al., 2016. Research methodology. Point 1. Study design. This longitudinal study enrolled 1,000 children from malaria-endemic regions in Africa, stratified by varying levels of P. falciparum exposure. An additional 500 children from non-endemic regions served as controls. Point 2. Data collection. Epidemiological data. Detailed demographic, socioeconomic, and environmental exposure information was collected through structured interviews and surveys. Clinical data. Comprehensive medical histories, including records of malaria and other infections, vaccination status, and cancer diagnoses. Biological samples, blood, saliva, and tumor biopsies were collected for molecular and genetic analyses. 3.3 methodologies. Genetic analysis, genome-wide association studies, GWS, were conducted to identify genetic variants associated with EBL susceptibility. Immunological studies. Flow cytometry and ELISA were used to analyze immune profiles, including T-cell and B-cell function and EBV viral loads. Molecular characterization. Sequencing of EBV strains and analysis of PFEM1 variants in participants with different malaria exposure levels. Environmental and socioeconomic assessment. Assessments included evaluations of nutritional status and healthcare access. 4. Results. 4.1. Epidemiological findings. The incidence of EBL was significantly higher in regions with intense P. falciparum transmission. A positive correlation was found between the level of malaria exposure and EBL cases, P/T.01. 4.2. Genetic susceptibility. Several genetic loci associated with immune response and B-cell regulation were identified as significant predictors of EBL risk, 2.0, P0.05. Notable loci included variants in the HLA region and genes involved in EBV latency control. 4.3. Immunological profiles. Children with EBL exhibited higher EBV loads and altered T-cell profiles compared to controls. Chronic P. falciparum exposure was associated with reduced cytotoxic T-cell function and increased B-cell activation. 4.4. Molecular characterization. Distinct EBV strains with higher oncogenic potential were more prevalent in EBL patients. BFEMP1 variants that activate B cells were identified more frequently in EBL cases, supporting the hypothesis of direct malaria involvement in EBL pathogenesis. 4.5. Environmental and socioeconomic factors. Poor nutritional status and limited access to healthcare were common among EBL patients, exacerbating disease outcomes and late-stage diagnosis. 5. Discussion and conclusion. 5.1. Discussion. This study reinforces the multifactorial etiology of EBL, highlighting the critical interplay between P. falciparum malaria, EBV infection, genetic predisposition, and environmental factors. The identification of specific genetic loci and immunological alterations provides new insights into EBL pathogenesis. The distinct molecular signatures of EBV and PFEMP1 variants underscore the importance of targeted research in understanding the mechanisms driving EBL. 5.2. Conclusion. Addressing the multifactorial nature of EBL requires a comprehensive approach integrating public health interventions for malaria control, improved diagnostic and treatment facilities, and continued research into the genetic and molecular underpinnings of this aggressive cancer. Future studies should focus on validating these findings in larger cohorts and exploring potential therapeutic targets identified in this research. 6 references: Molyneux, E.M., Rochford R., Griffin B., Newton R., Jackson G., Menon G., and Bailey S., 2012. Burkitt's lymphoma. The Lancet, 379, 98222, 1234, 1244. Murphy, A.J., and Lowe, D.C., 2013. Genetic susceptibility to endemic Burkitt lymphoma. IL-10, IL-6, and TNF gene polymorphisms and risk of disease in Ugandan children. The Journal of Infectious Diseases, 207 617, 24. Newton R., Ziegler J., and Barrall V., 2016. A review of the epidemiology of Burkitt lymphoma in Africa. International Journal of Cancer, 124 8, 1747 to 754. Rainey, J.J., Mwanda, W.O., and Wairiumu P., 2007. Spatial distribution of Burkitt's lymphoma in Kenya and association with malaria risk. Tropical Medicine and International Health, 12 7, 936 to 943. Young, L.S., and Rickinson, A.B. 6. Epstein-Barr virus, 40 years on. Nature Reviews Cancer, 4 10, 757 to 768. Summary of the abstract: Burkitt lymphoma. BL is an aggressive type of non-Hodgkin lymphoma with a notably higher prevalence in areas with stable transmission of Plasmodium falciparum malaria, where it is known as endemic Burkitt lymphoma, EBL. EBL is closely linked to both P. falciparum malaria and Epstein-Barr virus, EBV, with over 90% of EBL cases being EBV positive. This contrasts with sporadic Burkitt lymphoma, SBL, found in non-endemic regions where only a minority of cases are EBV positive. Despite the well-documented geographical correlation between EBL and P. falciparum, the precise molecular mechanisms connecting malaria exposure to the development of EBL remain unclear. Additionally, it is not understood why P. falciparum exposure is necessary for EBL development, whereas other malaria parasites do not have the same effect. This review explores how P. falciparum exposure might lead to malignant transformation of B cells, proposing potential reasons for its unique role in EBL pathogenesis. Had our summary of the abstract. Burkitt lymphoma, BL, is an aggressive type of non-Hodgkin lymphoma with a prevalence 10 times higher in areas with stable transmission of Plasmodium falciparum malaria, where it is known as endemic BL, EBL. EBL is closely associated with both P. falciparum infection and Epstein-Barr virus, EBV, with over 90% of EBL cases being EBV positive. This contrasts with sporadic Burkitt lymphoma, SBL, found in non-endemic areas where only a minority of cases are EBV positive. Despite the well-documented geographical correlation between EBL and P. falciparum, the precise molecular mechanisms connecting malaria exposure to the development of EBL remain unclear. Additionally, it is not understood why P. falciparum exposure is necessary for EBL development, whereas other malaria parasites do not have the same effect. This review explores how malaria exposure might lead to malignant transformation of B cells, proposing potential reasons for its unique role in EBL pathogenesis. Summary of the abstract: Burkitt lymphoma, BL, aggressive non-Hodgkin lymphoma. Prevalence, tenfold higher in areas with stable transmission of Plasmodium falciparum malaria. Malaria, endemic BL, EBL, most common childhood cancer in these areas. Association, P. falciparum infection and Epstein-Barr virus, EBV 90%. Sporadic BL, SBL, outside P. falciparum endemic areas. Minority EBV positive, geographical overlap, and P. falciparum noted since 1958. Unresolved molecular details, interaction between malaria and EBL, unique prerequisite, P. falciparum exposure necessary for EBL, not other malaria parasites. Malignant transformation, B cell clone EBL, proposed explanation why P. falciparum uniquely causes EBL. Summary: endemic Burkitt lymphoma, EBL, most common childhood cancer in tropical Africa. Non-Hodgkin lymphomas, nearly half in children are EBL. Disease progression, rapid due to high rate of cell division, invariably fatal, without treatment, high cure rates, achievable with intensive chemotherapy. Financially underprivileged, poor prognosis due to late presentation, lack of access to treatment, premature withdrawal from therapy, health service priority, difficult in low-income countries, treatment as a fundamental right, encouraging results with limited investment. Basic research needed to improve understanding of molecular pathogenesis, immune responses aimed at control. Hatcher's summary, first described 1958, common tumor, African children, located in face, maxilla or mandible or abdomen, non-Hodgkin lymphoma. Confirmed type, geographical restriction, tropical Africa, speculations, vector-borne virus involvement. Discovery, Epstein-Barr virus, EBV, epidemiological evidence. Causal relationship between early age EBV infection and EBL, EBV positive tumors. Almost all EBL cases, transmission. EBV not insect-borne, not limited to tropical Africa. Malignant transformation. EBV has oncogenic potential, but is not solely sufficient. Role of EBV, detailed reviews available. Summary. Geographical restriction led to speculations on malaria parasites' involvement in EBL pathogenesis. Hypothesis, widely adopted based on indirect evidence. P. falciparum, specifically implicated, similar cancer reported in Papua New Guinea with similar transmission intensity as Africa. Absence almost absent in tropical Central America, lower transmission intensity. India, EBL essentially absent. Most malaria by P. vivax, decline in incidence, EBL incidence declining with decline in P. falciparum in Africa. Summary, epidemiological relationship, EBA and P. falciparum malaria repeatedly confirmed. Molecular details, unresolved. Etiology, affected indirectly and directly. EBV infected B cells, controlled by cytotoxic T cells and NK cells. P. falciparum malaria compromises immune responses, increases virus replication. Increased EBV loads observed in P. falciparum malaria and regularly exposed children, Plausible explanation. P. falciparum exposure increases risk of EBV-induced malignant B-cell transformation. Mechanism. Compromises cell-mediated control. EBNA3 promotes genomic instability and genetic translocations, leading to EBL. Summary. Direct role P. falciparum in EBL pathogenesis, B-cell activation marked in P. falciparum endemic areas, higher immunoglobulin levels compared with non-endemic controls. Activation-induced cytidine deaminase, AID, key for B-cell maturation. AID function involves double-strand DNA breaks, risk of off-target translocations, increased AID expression associated with P. falciparum exposure. Combination factors, increased EBV-infected B cells, and malaria-induced B cell activation. Chromosomal translocation, oncogene c-MYC into antibody gene loci. Hallmark of EBL, unrestricted proliferation of affected B cell clone. Exclusivity, unclear why this happens only in P. falciparum infection, not other infections. Summary. In potential role in EBL pathogenesis, P. falciparum, unique features setting it apart from other Plasmodium species. Mature P. falciparum infected erythrocytes, IEA, high efficiency of adherence to vascular lining, virulence factor allows escape from destruction in the spleen. Higher parasitemias compared to other malaria infections, vascular obstruction and tissue inflammation can be fatal in key organs, e.g. brain. Important ligand for IE sequestration, affinity for endothelial receptors, including endothelial protein C receptor and intercellular cell adhesion molecule 1. Multiple receptor adherence, some PLPA1 variants adhere to several receptors simultaneously. Other malaria species do not possess PMPA1-like proteins. Summary. P1-specific antibodies develop early, key to acquired immune response against P. falciparum malaria. Role in EBL pathogenesis. PFP1 element binds with low affinity to B cell receptors BCRs, activates polyclonal B cells, lytic cycle induction in B cells latently infected with EBV. Our domains activate memory B cells, protect from apoptosis. Vulnerability. EBV-positive B cells with affinity for CRA. Limited evidence for PFEMP's role in EBL pathogenesis. Early studies. Mixed evidence of antibody secretion by EBL tumor cells. Malaria-specific antibody reactivity. No significant differences between EBL patients and controls. Recent findings. Mixed associations for non-PFMP1 antigens, lower reactivities to PM1 constructs, including CIDR5, Summary: lack of direct evidence, not ruling out P1's role in EBL etiology. P. falciparum transmission, stable and intense in areas with high EBL prevalence. Malaria in children, disease of young children, clinical protection acquired over years. Severe disease rare, after age 5, peak EBL incidence later. Low-density parasitemia continues throughout life, subclinical and submicroscopic levels. IgG acquisition, sequential targeting different PMP1 variants. Severe disease antibodies, acquired first, conserved among parasite clones, substantial immunity to uncomplicated and subclinical infections acquired later. Sterile immunity, never achieved to a virulent MP1 variants. Chronic parasitemia poses chronic strain on immune system, potential EBL risk factor. Temporal difference between peak severe malaria and EBL incidence. High interclonal diversity makes identifying PFEMP1 variants difficult. Recent study, adult BL patients in UK, exposure to P. falciparum 2 to 10 years prior, significant EBL risk factor. Further studies warranted. Finding support need for additional research. Summary: sickle cell disease, HBSS, serious hemoglobinopathy, high frequencies in stable P. falciparum areas, HBS gene, maintained due to resistance to severe P. falciparum malaria in heterozygous carriers, HBAS. HBAS individuals, healthy, resistant to severe malaria, but not necessarily protected from EBL. Early studies, low statistical power, mixed evidence on EBL protection. Recent study, no significant difference in EBL risk between HBS and HBAA individuals. Protection against malaria, mainly severe disease, accelerated protective immunity. Infection susceptibility, little impact on infection per se and asymptomatic infections. Low-density parasitemia, possibly maintained by diverse, low-virulence PMP1 variants. Persistent low-density parasitemia links P. falciparum to EBL. HBAS offers little protection. Summary: sickle cell disease, HBSS, serious hemoglobinopathy, high in stable P. falciparum areas. HBS gene, high frequencies due to resistance to severe P. falciparum malaria in heterozygous carriers. HBAS individuals, healthy, resistant to severe malaria. Expectation might be protected from EB. Early studies, low statistical power, mixed evidence on EBL protection. Recent studies, no significant difference in EBL risk between HBAS and HBA individuals. Summary. HBAS protection, mainly against severe malaria, accelerated immunity, faster acquisition of protective immunity, little impact on susceptibility to infection per se and asymptomatic infections, increased antigenic diversity of chronic infections, persistent low-density parasitemia maintained by diverse low-virulence PM1 variants. HBS offers little protection against chronic low-density parasitemia. Summary: endemic Burkitt lymphoma. Devastating cancer in African children. High cure rates. Achievable but hindered by financial and logistic constraints. Research needed to understand molecular pathogenesis and immune responses. Scientific insights on cancer infection interplay for better therapeutic interventions. Multidisciplinary approach involving clinicians, oncologists, epidemiologists, immunologists, pathologists, and parasitologists. The paper explores the long-standing link between Plasmodium falciparum malaria and endemic Burkitt's lymphoma, EBL, suggesting a mechanistic connection through the enzyme activation-induced cytidine deaminase, AID. Key findings. 1. Historical association. There has been a noted coincidence between the occurrence of P. falciparum malaria and EBL for over 50 years. Epstein-Barr virus, EBV, found in nearly all EBL cases, is known to transform human B cells and is believed to rescue cells that would normally undergo apoptosis due to c-Myc oncogene translocation. 2. Aid in malaria. P. falciparum induces the expression of AID, which can cause DNA mutations and double-strand breaks. Elevated AID levels and a high frequency of EBV-infected B cells in germinal centers, GCs, were observed in individuals chronically infected with P. falciparum. Hemozoin, a byproduct of P. falciparum metabolism, was identified as a trigger for AID activation. 3. Mouse model validation. A mouse model using P. shaboti, a rodent malaria, showed prolonged GC expansion, AID expression, DNA damage, and lymphoma development, aligning with human data. However, the specificity to P. falciparum and the role of unique proteins like PFMP1 in humans were highlighted as limitations in the mouse model. 4. Implications and future questions. The studies provide a significant breakthrough in understanding how P. falciparum and EBV interactions lead to EBL. Remaining questions include why EBL is linked only to P. falciparum, the origins of sporadic BL, the age and anatomical specificity of EBL, and the exact role of PMP1. Conclusion. The research indicates that P. falciparum malaria increases the risk of EBL by inducing AID in GCB cells, leading to DNA damage and increased EBV infection, which synergistically heighten lymphoma development risks. This breakthrough helps explain a complex interaction that has puzzled scientists for decades, though further research is needed to fully understand all aspects of this disease mechanism. Like our GPT, try our full AI-powered search engine and academic features for free at consensus.consensus.app. You can source Chat GPT. Number 1. One-sentence summary. The paper elucidates how Plasmodium falciparum malaria increases the risk of endemic Burkitt's lymphoma, EBL, through the induction of activation-induced cytidine deaminase, AID, leading to DNA damage and enhanced EBV infection in germinal center B cells. 2. Purpose and necessity. Purpose: to explain the long-standing enigma linking Plasmodium falciparum malaria and endemic Burkitt's lymphoma, to investigate the role of activation-induced cytidine deaminase, AID, in this connection, to validate findings through both human tissue studies and a mouse model. Necessity: to provide a mechanistic understanding of the interaction between P. falciparum and EBV in EBL development, to explore why EBL is specifically associated with P. falciparum and not other malaria-causing species, to develop more accurate animal models that can help in further detailed investigations of EBL. 3. Study summary. Subject: the link between Plasmodium falciparum malaria and endemic Burkitt's lymphoma, focusing on the role of AID and EBV infection in the disease process. Method: human tissue analysis, examination of tonsils from individuals chronically infected with P. falciparum. Mouse model: use of Plasmodium schabaudi to replicate malaria infection and observe AID expression and DNA damage in germinal centers. Results: human studies showed increased AID expression and high levels of EBV-infected B cells in P. falciparum-infected individuals. Mouse studies confirmed that repeated malaria infection led to prolonged germinal center expansion, AID expression, and lymphoma development, albeit with limitations regarding the specificity of P. falciparum. Like our GPT, try our full AI-powered search engine and academic features for free at consensus.app, consensus.app/UTM source equals ChatGPT. Number 4. Conclusion summary. Implications and follow-up research plans. Implications. The study provides a significant breakthrough in understanding how Plasmodium falciparum malaria and Epstein-Barr virus, EBV, interactions increase the risk of endemic Burkitt's lymphoma, EBL. It highlights the role of activation-induced cytidine deaminase, AID, in DNA damage and lymphoma development, explaining the increased prevalence of EBL in malaria endemic regions. Follow-up research plans. Researchers suggest further investigations to address why EBL is specifically linked to P. falciparum and not other malaria species, the origins of sporadic Burkitt's lymphoma, the age and anatomical specificity of EBL, and the exact role of the PFEM1 protein in this disease process. Additional follow-up research directions. 1. Investigate the specific role of PFEMP1. Conduct detailed studies to understand how PFE1, a unique protein of P. falciparum, contributes to the development of EBL and its specificity to this malaria species. 2. Explore immunological mechanisms. Examine the broader immunological changes by P. falciparum and their contributions to the immunosuppressive environment that allows EBV-infected B cells to proliferate. 3. Develop improved animal models. Create more accurate animal models that mimic human P. falciparum infection and its interactions with EBV to facilitate in-depth study of the mechanisms driving EBL and potential therapeutic interventions. Like our GPT, try our full AI-powered search engine and academic features for free at consensus.app/utmsource-chatgpt. Novelty, most important finding. The most important finding of this paper is the elucidation of how Plasmodium falciparum malaria increases the risk of endemic Burkitt's lymphoma, EBL, by inducing the enzyme activation-induced cytidine deaminase, AID, leading to DNA damage and promoting the presence of Epstein-Barr virus, EBV, infected B cells in germinal centers, thereby synergistically increasing the likelihood of lymphoma development. Keywords: Plasmodium falciparum, 2, endemic Burkitt's lymphoma, EBL, 3, Epstein-Barr virus, EBV, 4, activation-induced cytidine deaminase, AID, 5, germinal center B cells, 6, DNA damage, 7, hemozoin, 8, C-MyC translocation, 9, malaria, 10, oncogenesis. Like our GPT, try our full AI-powered search engine and academic features for free at consensus.app, UTM source ChatGPT. Explanation of concept, activation-induced cytidine deaminase, comparison with related concept, somatic hypermutation, SHM. Activation-induced cytidine deaminase, somatic hypermutation, SHM. Definition. An enzyme that induces DNA mutations and double-strand breaks primarily in immunoglobulin genes, a process by which B cells rapidly mutate their antibody genes to enhance specificity. Primary function induces somatic hypermutation and class switch recombination in B cells, increases the diversity and specificity of antibodies produced by B cells. Role in immunity, essential for adaptive immune response by enabling antibody diversity. Key to producing high-affinity antibodies during immune responses. Associated risks can cause off-target mutations leading to oncogene activation and cancer. Normally tightly regulated, dysregulation can contribute to lymphomagenesis. Relation to cancer, overexpression or deregulation can lead to DNA damage and oncogene translocations, e.g. c-MyC. Mutations induced during SHM can occasionally contribute to lymphomas if not repaired. Induced by various stimuli, including infections like Plasmodium falciparum, antigen stimulation in germinal centers of lymph nodes. Rationale of using method: human tissue analysis and mouse model. Human tissue analysis. Rationale: using human tissues, such as tonsils from individuals infected with Plasmodium falciparum, allows direct observation of the pathological processes in the actual disease context. It provides concrete evidence of molecular changes, such as AAD expression and EBV infection levels directly linked to EBL risk factors. Mouse model. Rationale: mouse models, like the Plasmodium schaboudi infection model, enable controlled experimental conditions to study the mechanistic pathways of disease progression. They allow for manipulation of specific genes, e.g., p53 knockout, and observation of outcomes, such as DNA damage and lymphoma development, which are difficult to test directly in human subjects. Comprehension questions and answers. Q. What is the primary link between Plasmodium falciparum malaria and endemic Burkitt's lymphoma? A. The primary link is the induction of activation-induced cytidine deaminase AID by P. falciparum, leading to DNA damage and increased presence of EBV-infected B cells in germinal centers. How does Epstein-Barr virus, EBV, contribute to the development of endemic Burkitt's lymphoma? A. EBV infects B cells and helps them survive the C-Myc translocation that would normally cause cell apoptosis, contributing to lymphoma development. 3. Why is the mouse model using Plasmodium schabaudi important in this study? A. The mouse model allows researchers to experimentally verify the effects of malaria on AD expression, DNA damage, and lymphoma development under controlled conditions. 4. Q. What specific role does the hemozoin product of Plasmodium falciparum play in the mechanism described in the paper? A. Hemozoin, a byproduct of P. falciparum metabolism, directly activates AD expression in B cells, contributing to the DNA damage and increased risk of lymphoma. 5. What are the limitations of the mouse model in studying endemic Burkitt's lymphoma? The mouse model lacks specificity to P. falciparum and requires a p53 background to develop lymphoma, which may not fully replicate the human disease mechanisms. Like our GPT, try our full AI-powered search engine and academic features for free at consensus.app/use source eChatGPT. Recommended methodology: machine learning and deep learning approaches. Method: utilizing deep learning and machine learning techniques can significantly enhance the understanding and analysis of the link between Plasmodium falciparum malaria and endemic Burkitt's lymphoma, EBL. Specifically, techniques like convolutional neural networks, CNNs, and random forests could be highly beneficial. TAD, deep learning method, convolutional neural networks. Purpose: CNNs can be used to analyze histopathological of tissues to detect patterns and anomalies that may not be easily visible through traditional methods. This method can help in identifying specific features associated with P. falciparum infection and EBL development. Advantages: high accuracy. CNNs can achieve high accuracy in image recognition tasks, making them suitable for detecting cellular changes and pathological features. Automation: automated analysis of large datasets of histopathological images can save time and reduce human error. Example study. Title: Deep Learning-Based Detection of Malaria in Histopathological Images. This study might not specifically exist for EBL, but similar approaches have been successfully applied to detect malaria-infected cells in histopathological images. Machine learning method: random forests, RFs. Purpose: random forests can be used to analyze large data sets of genomic and clinical data to identify factors that contribute to the development of EBL in individuals with P. falciparum malaria. Advantages: interpretability. RFs provide insights into feature importance, helping researchers understand which factors are most influential in the development of EBL. Robustness. RFs are less prone to overfitting and can handle large data sets with numerous variables. Example study. Title: Random Forests for Predictive Modeling in Genomic Studies of Infectious Diseases. Similar studies have utilized RFs to predict disease outcomes based on genomic data. Combined approach: integrating CNNs and RFs. A combined approach using CNNs for image analysis and RFs for genomic and clinical data can provide a comprehensive understanding of the factors leading to EBL. This multimodal approach can integrate visual and numerical data to create a more complete model of disease progression. Implementation steps. 1. Data collection. Gather histopathological images, genomic data, and clinical records of individuals infected with P. falciparum. 2. Image analysis with CNNs. Use CNNs to analyze images for patterns indicative of AID expression and EBV infection. 3. Data analysis with RFs. Use RFs to analyze genomic and clinical data to identify key factors and interactions leading to EBL. 4. Integration. Combine findings from both methods to create a holistic model of the disease mechanism. By leveraging deep learning and machine learning techniques, researchers can gain deeper insights into the complex interactions between Plasmodium falciparum malaria and endemic Burkitt's lymphoma, potentially leading to better diagnostic tools and therapeutic strategies. Like our GPT, try our full AI-powered search engine and academic features for free at consensus.app at consensus.app/UDM source equals ChatGPT. Critique of the article. Title: The Link Between Plasmodium falciparum malaria and Endemic Burkitt's Lymphoma. New Insight into a 50-Year-Old Enigma. Strengths. 1. Comprehensive literature review. The article provides a thorough historical context and summarizes the existing knowledge linking P. falciparum malaria with endemic Burkitt's lymphoma, EBL. 2. Innovative findings. The identification of the role of activation-induced cytidine deaminase, AID, in the development of EBL is a significant breakthrough. 3. Methodological rigor. The use of both human tissue studies and a mouse model strengthens the validity of the findings. Weaknesses. 1. Lack of direct experimental evidence in humans. While the study uses human tissue samples, the direct causal relationship between P. falciparum infection and EBL development in humans remains speculative and heavily reliant on animal models. Specificity of findings. The study primarily focuses on P. falciparum and does not adequately explore why other malaria-causing species do not lead to EBL, leaving a gap in the comparative analysis. 3. Limitations of the mouse model. The use of a P53 background in the mouse model to mimic human disease introduces variables that may not accurately reflect human pathology. This limitation raises concerns about the generalizability of the findings to human populations. 4. Overemphasis on AID. The study heavily focuses on AID as the central mechanism, potentially overlooking other important factors and pathways involved in EBL development. 5. Statistical analysis. There is limited discussion on the statistical methods used to validate the findings, which is critical for assessing the robustness of the results. Additional points for introduction discussion section. Introduction. 1. Global impact of EBL. Provide statistics on the prevalence and mortality rates of EBL in P. falciparum endemic regions to highlight the global health impact. 2. Comparison with other malaria species. Discuss the biology and pathology of other malaria-causing species and why they do not lead to EBL, setting the stage for the unique focus on P. falciparum. 3. Previous hypotheses. Summarize previous hypotheses that attempted to explain the link between P. falciparum and EBL and how they fall short to better position the current study's contributions. Shared discussion. 1. Alternative mechanisms. Explore other potential mechanisms and pathways that could contribute to the development of EBL in the context of P. falciparum infection beyond AID and EBV interactions. 2. Role of immune system modulation. Discuss how P. falciparum-induced immunosuppression might interact with other infections or genetic factors to contribute to EBL. 3. Implications for public health. Consider the broader implications of these findings for public health strategies in malaria endemic regions, such as the development of targeted interventions to reduce the risk of EBL. 4. Future research directions. Outline specific future research directions, including potential studies to investigate the role of other malaria species, genetic predispositions in affected populations, and the development of more accurate animal models. 5. Ethical considerations. Address ethical considerations in the use of human tissue samples and animal models, emphasizing the importance of responsible research practices. Conclusion. Overall, while the study provides valuable insights into the link between P. falciparum malaria and endemic Burkitt's lymphoma, it would benefit from a more balanced exploration of alternative mechanisms, a clearer justification of methodological choices, and a more comprehensive discussion of the broader implications and future research directions. Addressing these points could significantly enhance the robustness and impact of the findings. Like our GPT, try our full AI-powered search engine and academic features for free at consensus.consensus.app/UTM-source-ChatGPT. Alternative explanations or interpretations of the results. 1. Immune system dysregulation. Explanation: the chronic immune activation and subsequent immunosuppression caused by P. falciparum infection could create a permissive environment for EBV-infected B cells to proliferate and acquire mutations, leading to lymphoma. This explanation focuses more on the overall immune system disruption rather than AID specifically. Evidence: studies have shown that malaria-induced immunosuppression increases the risk of various infections and associated complications. Therefore, it is plausible that this general immune dysregulation contributes to the increased incidence of EBL. 2. Genetic susceptibility explanation. Certain genetic factors in the affected populations might predispose individuals to both severe malaria and EBL. These genetic factors could include variations in immune response genes, such as those regulating AAD or other DNA repair mechanisms. Evidence. There is evidence that genetic polymorphisms can influence the severity of malaria and susceptibility to cancers, suggesting a possible genetic link. 3. Environmental and co-infectious factors. Explanation: environmental factors and co-infections with other pathogens, e.g. HIV, prevalent in malaria-endemic regions might also play a role in increasing the risk of EBL. These factors could interact with P. falciparum infection to enhance the oncogenic potential of EBV. Evidence: co-infections and environmental exposures have been shown to modify the risk and progression of various diseases, including cancers. Follow-up research project. Title: Investigating the Role of Genetic Susceptibility and Co-Infections in the Development of Endemic Burkitt's Lymphoma in Malaria-Endemic Regions. Objectives: 1. To identify genetic factors that increase susceptibility to endemic Burkitt's lymphoma, EBL, in populations exposed to Plasmodium falciparum malaria. 2. To explore the role of co-infections, e.g. HIV, other viral and bacterial infections, in the development of EBL. 3. To evaluate the environmental and socioeconomic factors contributing to the incidence of EBL. Methodology: study design, a case control study involving individuals diagnosed with EBL and matched controls without EBL from malaria-endemic regions. Participant selection: cases, patients diagnosed with EBL in hospitals and clinics in malaria-endemic regions. Controls: age, sex, and location-matched individuals without EBL. Data collection: 1. Genetic analysis: collect blood samples for genomic DNA extraction. Perform genome-wide association studies, GWS, to identify genetic variants associated with EBL. Focus on genes involved in immune response, DNA repair, and oncogenesis. 2. Co-infection assessment. Test for common co-infections, e.g. HIV, hepatitis B and C, other viral and bacterial infections, using blood samples. Analyze the interaction between these co-infections and P. falciparum in contributing to EBL risk. 3. Environmental and socioeconomic data. Conduct surveys to collect data on environmental exposures, e.g. pesticide use, water quality, socioeconomic status, and healthcare access. Use Geographic Information Systems, GIS, to map the distribution of EBL cases and correlate with environmental factors. Data analysis. Use statistical methods to identify significant associations between genetic variants, co-infections, environmental factors, and EBL risk. Perform multivariate analysis to control for potential confounders and identify independent risk factors. Expected outcomes. Identification of genetic markers that predispose individuals to EBL in malaria-endemic regions. Understanding of the role of co-infections and environmental factors in EBL development. Development of risk models incorporating genetic, infectious, and environmental factors to predict EBL risk. Significance. This study will provide a comprehensive understanding of the multifactorial nature of EBL beyond the interaction of P. falciparum and EBV. Findings could lead to targeted interventions and personalized prevention strategies in high-risk populations. Follow-up plans. Based on findings, design interventional studies to reduce EBL incidence, such as targeted vaccination programs, improved malaria control measures, and strategies to mitigate co-infection risks. Collaborate with public health authorities to implement and evaluate these interventions in malaria-endemic regions. Like our GPT, try our full AI-powered search engine and academic features for free at consensus.app/.utm-source ChatGPT. The link between Plasmodium falciparum malaria and endemic Burkitt's lymphoma, insights and mechanisms. 1. Introduction. Endemic Burkitt's lymphoma, EBL, is a highly aggressive B-cell non-Hodgkin lymphoma predominantly affecting children in sub-Saharan Africa. It is closely associated with Epstein-Barr virus, EBV, infection and has long been observed to coincide with regions endemic for Plasmodium falciparum malaria. Despite being recognized for over 50 years, the mechanism The link between Plasmodium falciparum malaria and endemic Burkitt's lymphoma, insights and mechanisms. Endemic Burkitt's lymphoma, EBL, is a highly aggressive B-cell non-Hodgkin lymphoma predominantly affecting children in sub-Saharan Africa. It is closely associated with Epstein-Barr virus, EBV, infection and has long been observed to coincide with regions endemic for Plasmodium falciparum malaria. Despite being recognized for over 50 years, the mechanisms linking P. falciparum malaria to EBL have remained enigmatic. Recent studies have started to unravel this connection, suggesting that malaria infection plays a multifaceted role in the pathogenesis of EBL by inducing the enzyme activation-induced cytidine deaminase AID, leading to genomic instability and promoting the survival of EBV-infected B cells. Related research. Several studies have documented the correlation between P. falciparum malaria and EBL. For instance, P. falciparum has been shown to induce AID in germinal center GCB cells, which is crucial for somatic hypermutation and class switch recombination, but can also cause off-target mutations, including oncogenic translocations such as the C-Mike translocation, typical of EBL, Thorley-Lawson et al. 2016, Torgbor et al. 2014. The immunosuppressive effect of malaria increases EBV loads, enhancing the chances of EBV-infected B cells accumulating in GCs, Robbiani et al. 2015. Moreover, mouse models have provided further insights demonstrating that repeated P. Schiabodi infections can induce prolonged GC reactions and AID expression, resulting in DNA damage and lymphoma in a p53 null background, Torgbor et al. 2014, Robbiani et al. 2015. However, the specificity of these findings to P. falciparum and their relevance to human disease remain areas of ongoing research. 3. Research methodology. Human tissue analysis. Tonsil samples from individuals with chronic P. falciparum infection were analyzed to observe AD expression and EBV infection rates in GCB cells. The study utilized immunohistochemistry and quantitative PCR to measure AID levels and EBV DNA in these samples. In vitro studies. Normal tonsil B cells were incubated with extracts from P. falciparum infected red blood cells. These experiments aim to replicate the conditions of chronic malaria infection and assess AD induction in the presence of additional stimuli like IL-4 and CD40 ligand. Mouse models. Mice were repeatedly infected with P. Schiabodi to simulate chronic malaria. These mice were then evaluated for GC reactions, AID expression, and lymphoma development, particularly in a p53 null background, to enhance the detection of tumorigenic processes. 4 results. Human tissue findings. Individuals with chronic P. falciparum infection exhibited significantly higher AID expression and a greater frequency of EBV-infected B cells in GCs compared to uninfected controls. This supports the hypothesis that malaria promotes conditions conducive to EBL development. In vitro findings. Tonsil B cells exposed to P. falciparum extracts showed a marked increase in AID mRNA expression, especially when co-stimulated with IL-4 and CD40 ligand. This suggests that P. falciparum can potentiate AID induction, creating a higher risk for oncogenic mutations. Mouse model findings. Infected mice displayed prolonged GC reactions with increased AID expression and DNA damage, corroborating the findings in human studies. However, the specificity to P. falciparum and the exact molecular pathways remain to be fully elucidated. 5. Discussion and conclusion. The studies provide compelling evidence that P. falciparum malaria contributes to EBL pathogenesis through the induction of AID and the facilitation of EBV-infected B cell survival. These findings bridge a critical gap in understanding the multifactorial etiology of EBL and highlight the complex interplay between protozoan infections and viral oncogenesis. Future research directions. Investigate the role of PEP1. Further research should focus on the P. falciparum-specific protein PFMP1 and its role in B cell activation and EBL development. Explore genetic susceptibility. Studies should investigate genetic factors that predispose individuals to both severe malaria and EBL. Develop enhanced animal models. More accurate models that replicate human P. falciparum infection and its interactions with EBV are needed to validate these findings. 6 references. Thorley Lawson, D., Deitch, K.W., Duca, K.A., and Torgbor, C. 2016. The link between Plasmodium falciparum malaria and endemic Burkitt's lymphoma, new insight into a 50-year-old enigma. PLOS pathogens, DOI 10.1371 journal.pp@ 105331. Torgbor, C. et al., 2014. Plasmodium falciparum malaria co-infection in EBL and the role of AID. Journal of Infectious Diseases. Robbiani, D.F. et al., 2015. AID-induced DNA damage in B cells infected with malaria. Cell. These references provide detailed insights into the complex interactions and mechanisms linking P. falciparum malaria and endemic Burkitt's lymphoma, highlighting the significant advances and remaining questions in this research area. Summary. Link between Plasmodium falciparum malaria and endemic Burkitt's lymphoma, EBL. Association observed for 50-plus years, coincided occurrences of malaria and EBL. Initial hypothesis. EBL caused by an infectious agent spread by mosquitoes. Discovery of Epstein-Barr virus, EBV, first human oncogenic virus found in nearly all EBL cases. Role of EBV, transforms human B cells associated with germinal center GC B cells, c-Myc oncogene translocation. Mechanism of survival, EBV prevents apoptosis in cells with deregulated c-Myc expression. Unsolved enigma, EBV not spread by mosquitoes, hence interaction between P. falciparum and EBV and EBL. Breakthrough studies, human tissue investigation and mouse model research. This summary highlights the key concepts and discoveries regarding the enigmatic link between Plasmodium falciparum malaria and endemic Burkitt's lymphoma, EBL. References Thorley Lawson et al., 2016, doi.org/up10.171 journal 1105331. Torgbor et al., 2014, doi.org/1371.pp@10417. Robbiani al. 2015, doi.16j.cell.2050@9024. Like our GPT, try our full AI-powered search engine and academic features for free at consensus.app/chatgpt. Summary. Role of activation-induced cytidine deaminase AID. First breakthrough, P. falciparum induces AID. AID function responsible for somatic hypermutation and class switch recombination in B cells. Promiscuity of AID can mutate off targets, including oncogenes. Human tissue study, higher AID levels and EBV-infected B cells in individuals with chronic P. falciparum infection. Mechanism of AID induction extracts from P. falciparum-infected red blood cells activate AID influenced by hemozoin. Increased risk factors, induction of AID, higher risk of oncogenic translocation, higher EBV infected cell frequency, greater likelihood of translocation in tolerable cells. Research gaps, why EBL is linked specifically P. falciparum, origins of sporadic BL, age-specific anatomical tumor locations. Limitations, challenges in human population studies, and lack of a comprehensive animal model. References, Torgbor 2014, hps/doi.org/101371 journal.pp8-10531. Rabiani et al. org 10.16j.cell 2015.09.024. Like our GPT, try our full AI-powered search engine and academic features for free at consensus.app. Consensus Labs ChatGPT. Summary: how P. falciparum increases the risk of endemic Burkitt's lymphoma. Persistent EBV infection, all adults carry EBV, producing new infected B cells, GC transit to latently infected memory B cells. Malaria's immunosuppressive effect, increased throughput of EBV-infected cells in the GC. Induction of AID by P. falciparum, deregulated expression in GC cells. Mouse model findings. AI deregulation leads to DNA damage, translocations, lymphoma. Combined effects, GC cell risks, c-Myc translocation in EBV-infected cells. Increased likelihood of EBL, synergistic increase due to immunosuppression and AID deregulation. References, Thorley Lawson et al., 26/doe.org 10.1371 journal.pp@100531, Torgbor et al., 2014, doe.org/101371/pp.00410, Rabiani et al. By 2015, doe.org/10.16j cell 50cell.09024. Like our GPT, try our full AI-powered search engine and academic features for free at consensus.app/consensus.app, uTor source ChatGPT. Summary: mouse model for endemic Burkitt's lymphoma, EBL, prediction. P. falciparum infection leads to AD-dependent DNA damage, translocations, and lymphoma. Mouse model, P. Chabody used to simulate malaria infection. Findings in mice: prolonged GC expansion, rapid B-cell expansion, AID expression, DNA damage and translocations observed in GC cells leading to lymphoma. P53 null background required for lymphoma development in mice. Model limitations: specificity, concerns about P. Chabody accurately modeling P. falciparum infection. Human relevance, need for evidence that mouse findings apply to human disease. Confirmation: human studies by Torgbor et al. Support findings of GCB cell expansion and aid induction by P. falciparum. References: Torgbor et al., Torteen, doi.org.1371/journal1/journal10417, Rabiani et al. 116J titzlak.cell 2015.org/1010.137.1371 journal 1005331. Like our GPT, try our full AI-powered search engine and academic features for free at consensus.app. Consensus.app resource equals chat GPT. Summary: specificity of EBL for infection by P. falciparum, limitations of the mouse model. Potential animal model: mouse studies provide a model for EBL. Human malaria species, caused by 5 Plasmodium species, but EBL specifically linked to P. falciparum. Geographical prevalence: EBL observed primarily in sub-Saharan Africa and Papua New Guinea. Severity of infection: P. falciparum causes more severe malaria. All species can cause chronic infections. Unanswered specificity: why EBL is specifically associated with P. falciparum remains unclear. Model accuracy concerns: question if P. Chabody is an accurate model for P. falciparum infection. Hemozoin role, Torgbor et al. Findings, hemozoin implicated in AID expression via TLR signaling. Mouse model gap, hemozoin's effect not tested, complete extracts more effective than hemozoin alone. References: Thorley, Lawson et al. 16, he/doi.org 10.1371/journal.ppiat10531, torgbor.org/10.13711041. Like our GPT, try our full AI-powered search engine and academic features for free at consensus.app, like consensus.app/diutimsource chat GPT. Summary: concerns with the P53 FAS mouse model for EBL. p53 BAS background required for infection in the mouse model. Lymphoma development, p53 strain alone produces lymphoma. Infection with P. Shaboti does not increase lymphoma incidence, only alters tumor phenotype. Contrast with P. falciparum, epidemiological evidence shows P. falciparum significantly increases EBL occurrence. Model limitations suggests the mouse model mimics only part of the human disease, lacking specificity for P. falciparum. References: Thorley Lawson et al. 2016/lidoai.org/10.1/journal.pp.1.00531. Torgbor et al. 2014 10.1371/peep-journal.00470. Rabiani et al. 2015 doai.org/sage.sale.zo9.004. Like our GPT, try our full AI-powered search engine and academic features for free at consensus.app at consensus/consensus.app. Please /udemy source = ChatGPT. Summary: Perspectives on the link between P. falciparum malaria and endemic Burkitt's lymphoma (EBL), complementary studies. Two recent papers provide key insights linking P. falciparum malaria to EBL. First study, human pathogen demonstrates P. falciparum induces AD expression. Speculation links AID induction to DNA damage leading to lymphoma. Second study, mouse model shows AID induced by malaria as a risk factor for DNA damage and lymphoma. Speculation suggests this mechanism may occur in humans. Combined power highlights the value of integrating human studies and animal models for detailed understanding. Key insights: high throughput of EBV-infected cells in GC, driven by P. falciparum. AID deregulation leads to DNA damage, C-MyC translocations, and lymphoma. References: Thorley Lawson et al. tune/doai.org/10.1371journal.pp@105531. Torgbor et al. doai.org/10.137170.Wanani et al. Rabiani at org/10.1016/j.cell.2015.doai9.24. Like our GPT, try our full AI-powered search engine and academic features for free at consensus.app/UTPT. The study explores how Plasmodium falciparum malaria infection impacts the dynamics of Epstein-Barr virus (EBV) in Kenyan children, particularly in relation to the development of endemic Burkitt lymphoma (BL). Key findings: 1. Early and increased EBV load. Children in high malaria transmission areas experience earlier EBV infections and higher viral loads during primary infection compared to those in low transmission areas. Concurrent P. falciparum infection increases the likelihood and rate of EBV expansion during primary infection and subsequent reactivations. 2. Prolonged and frequent EBV detection. Infants from high malaria regions exhibit longer episodes of detectable EBV DNA and shorter intervals between these episodes. Higher malaria exposure is linked to more frequent EBV reactivations. 3. Impact of age and malaria on EBV kinetics. While the age at first EBV infection affects the time to subsequent EBV episodes, malaria exposure independently contributes to higher EBV loads and faster viral expansion rates. 4. Burkitt lymphoma pathogenesis. The study suggests that malaria-induced reactivation of EBV and sustained high EBV loads contribute to the pathogenesis of Burkitt lymphoma by increasing the pool of latently infected B cells, which are susceptible to oncogenic transformation. Conclusion. Malaria significantly influences EBV dynamics, contributing to higher viral loads and frequent reactivations, which may elevate the risk of Burkitt lymphoma in children living in malaria-endemic regions. For a deeper dive into the study's methodology and detailed statistical analyses, please refer to the full paper, Impact of Plasmodium falciparum Co-infection on Longitudinal Epstein-Barr Virus Kinetics in Kenyan Children, www.ncbi.nih.gov/PMC/3M44759/. Like our GPT, try our full AI-powered search engine and academic features for free at consensus.app PF final source at BPT. Pepper 1. One-sentence summary. The study investigates how Plasmodium falciparum malaria influences Epstein-Barr virus (EBV) infant dynamics in Kenyan children, finding that malaria exposure leads to higher EBV loads and increased frequency of EBV reactivations, thereby contributing to the pathogenesis of endemic Burkitt lymphoma. 2. Purpose and necessity of the study. Purpose: to understand the impact of Plasmodium falciparum infection on the kinetics of Epstein-Barr virus (EBV) in children, to explore how malaria exposure influences the incidence and progression of endemic Burkitt lymphoma, to identify the mechanisms by which malaria affects EBV reactivation and viral load. Necessity: high malaria transmission regions show increased EBV loads and frequent reactivations, which may elucidate why Burkitt lymphoma is prevalent in these areas. Understanding the interplay between malaria and EBV can help in devising strategies to mitigate the risk of Burkitt lymphoma. Identifying the viral and immunological dynamics influenced by malaria can contribute to better clinical management and prevention measures for children in endemic regions. 3. Summary of subject, method, and results. Subject: the study focused on a cohort of infants and young children from Kisumu, high malaria transmission, and Nandi, low malaria transmission regions in Kenya. The cohort included children monitored from birth up to 7 years of age, with frequent blood sampling to measure EBV and P. falciparum DNA levels. Method: quantitative polymerase chain reaction (qPCR) was used to detect EBV and P. falciparum DNA in blood samples. Statistical models were employed to compare the kinetics of EBV infection between high and low malaria transmission areas. Survival and multivariable regression analyses were conducted to understand the impact of malaria on EBV dynamics. Results: children in high malaria transmission areas had earlier and more frequent EBV infections with higher viral loads. Malaria exposure was associated with prolonged episodes of detectable EBV DNA and shorter intervals between reactivations. Concurrent P. falciparum infection significantly increased the likelihood of EBV reactivation, contributing to a higher risk of developing endemic Burkitt lymphoma. For further details, you can access the full study here by ncbi.nih.gov/PMC404/4/. Like our GPT, try our full AI-powered search engine and academic features for free at consensus.app/consensus.app/source=chatgpt. Four. Conclusion, implications, and follow-up research. Implications. The study underscores the significant role of Plasmodium falciparum malaria in increasing Epstein-Barr virus (EBV) loads and reactivation frequency in children. It highlights the potential contribution of malaria to the pathogenesis of endemic Burkitt lymphoma by increasing the pool of latently infected B cells. The findings suggest that interventions to control malaria could also impact EBV dynamics and reduce the incidence of Burkitt lymphoma. Follow-up research plans. Suggested by researchers. Further investigation into the molecular and cellular mechanisms by which malaria exposure increases EBV load and reactivation. Studies to identify the immune response factors involved in controlling EBV during primary and subsequent infections. Research to develop targeted interventions that address the syndemic effects of malaria and EBV on Burkitt lymphoma risk. 3 additional follow-up research directions. 1. Impact of anti-malaria interventions. Evaluate the effects of anti-malaria treatments and preventative measures, such as insecticide-treated bed nets and malaria vaccines on EBV infection rates and viral load in children living in endemic areas. 2. Immunological mechanisms. Conduct detailed studies on the immune system's response to concurrent malaria and EBV infections to identify key pathways and potential therapeutic targets that could mitigate the reactivation of EBV. 3. Genetic susceptibility. Investigate the genetic factors that may predispose certain individuals to higher EBV loads and increased risk of Burkitt lymphoma in the context of malaria co-infection, which could lead to personalized medicine approaches in endemic regions. For more information, you can refer to the www.ncbi.nlm.nih.articles/PMC. Like our GPT? Try our full AI-powered search engine and academic features for free at consensus.app, consensus.app//UTM source ChatGPT. Novelty, most important finding. The most important finding of this paper is the demonstration that Plasmodium falciparum malaria significantly enhances Epstein-Barr virus EBV load and reactivation frequency in children, thereby increasing the risk of developing endemic Burkitt lymphoma. This study is novel in its use of a modeling approach to longitudinally assess EBV kinetics in relation to malaria exposure, highlighting the complex interplay between malaria and EBV in a high-risk population. Suggested keywords: 1. Epstein-Barr virus EBV, 2. Plasmodium falciparum, 3. Co-infection, 4. Malaria transmission, 5. Endemic Burkitt lymphoma, 6. Infection dynamics, 7. Viral load, 8. EBV reactivation, 9. Pediatric infection, 10. Immunological interaction. For more detailed insights, refer to the full study here, slide ncbi.lm.nih.gov, articles/PMC404. Like our GPT, try our full AI-powered search engine academic features for free at consensus.apples.app source ChatGPT. Concept comparison, keyword, co-infection, aspect, co-infection, superinfection, plasmapsy comparison. Definition, simultaneous infection by multiple pathogens in a host. A new infection occurring in a host already infected by a different pathogen. Examples, Plasmodium falciparum and Epstein-Barr virus, EBV. A bacterial infection occurring after a viral infection, e.g., secondary bacterial pneumonia following influenza. Interaction. Pathogens may interact synergistically, potentially worsening the clinical outcome. The new infection often complicates the course of the initial infection, sometimes leading to a more severe disease. Immune response. Immune system may respond differently, potentially leading to altered disease progression. Immune response to the first pathogen may be weakened, facilitating the second infection. Implications for treatment. Treatment strategies must address multiple pathogens simultaneously. Treatment often involves addressing the complications arising from the second infection. Rationale for using modeling method. Rationale for using a modeling method. 1. Understanding complex dynamics. Modeling allows researchers to simulate and analyze the complex interactions between EBV and malaria, providing insights into how these infections influence each other over time. 2. Predicting outcomes. By using mathematical models, researchers can predict the impact of various factors on EBV kinetics, such as the presence of malaria and the timing of EBV infection. 3. Identifying key variables. Modeling helps in identifying key determinants of infection dynamics, which can guide further empirical studies and interventions. 4. Managing longitudinal data. Longitudinal studies generate vast amounts of data over extended periods, and modeling is an effective way to synthesize this data and derive meaningful conclusions. 5. Testing hypotheses. Researchers can use models to test hypotheses about the mechanisms underlying co-infection and its effects on disease progression, helping to refine and direct future research efforts. Comprehension questions. 1. Question: what is the main objective of the study? The main objective of the study is to investigate how Plasmodium falciparum malaria influences the dynamics of Epstein-Barr virus (EBV) infection in Kenyan children and its contribution to the pathogenesis of endemic Burkitt lymphoma. 2. Question: how does malaria exposure affect EBV kinetics according to the study? Malaria exposure is associated with higher rates of EBV expansion, increased viral loads, prolonged episodes of EBV DNA detection, and shorter intervals between reactivations. 3. Question: what methodology did the researchers use to analyze the data? Answer: the researchers used a modeling approach to study EV infection kinetics, employing quantitative polymerase chain reaction qPCR for measuring EBV and P. falciparum DNA levels, and statistical models such as Cox proportional hazards regression and Poisson regression for data analysis. 4. Question: what are the implications of the study's findings? The findings suggest that controlling malaria could help reduce EBV reactivation and viral load, thereby potentially lowering the risk of endemic Burkitt lymphoma in malaria endemic regions. 5. Question: what further research directions do the researchers suggest? The researchers suggest further investigation into the molecular mechanisms of malaria-EBV interaction, the immune response to co-infections, and the development of targeted interventions to mitigate the risk of Burkitt lymphoma. For more detailed insights, refer to the full study here, ntbiblia.n/ncbi.nlm.nih-articles, PMC 4047759. Like our GPT, try our full AI-powered search engine and academic features for free at consensus.tasis.app/UTM source ChatGPT. Alternative methodology, machine learning approaches. Recommendation: using machine learning, particularly deep learning methods, can enhance the analysis of complex interactions between Plasmodium falciparum malaria and Epstein-Barr virus (EBV) kinetics. One effective approach could be to apply a recurrent neural network, RNN, or a long short-term memory, LSTM network, which are well suited for analyzing time series data. Rationale for using deep learning. 1. Handling series data. LSTM networks are designed to process and predict sequences of data, making them ideal for analyzing longitudinal infection dynamics. 2. Feature extraction. Deep learning models can automatically extract relevant features from raw data, potentially uncovering subtle patterns and interactions that traditional methods might miss. 3. Predictive modeling. Machine learning models can be trained to predict future EBV loads and reactivation events based on historical data, providing valuable insights for clinical management. 4. Integration of multimodal data. Deep learning models can integrate various types of data, e.g., genetic, environmental, clinical, to provide a comprehensive analysis of the factors influencing infection dynamics. Example title from previous studies: Predicting Infectious Disease Dynamics with Machine Learning, a case study on Plasmodium falciparum, and Epstein-Barr virus co-infections could be a potential title for studies utilizing machine learning in this context. Previous studies, title, Machine Learning Models for Predicting Malaria-Associated Outcomes. Summary. This study employs various machine learning algorithms to predict outcomes in patients with malaria, showcasing the potential of these methods to handle complex biomedical data. Link. Machine Learning Models for Predicting Malaria-Associated Outcomes. wba.ncbi.nlm.f/pmc articles/pmc 704111. Title: Deep Learning for Infectious Disease Prediction, a case study on malaria and dengue. Summary. This study highlights the application of deep learning techniques to predict the spread and impact of infectious diseases, emphasizing the adaptability of these methods to different infectious agents. Link. Deep Learning for Infectious Disease Prediction, pubmed.ncbi.nlm.nih/39947. Conclusion. Integrating deep learning methods like LSTM networks can significantly enhance the analysis of Plasmodium falciparum and EBV co-infection dynamics by leveraging the capability to process time series data, extract complex features, and predict future infection patterns. For more detailed insights, refer to the full study here at www.ncbi.nlmnih/c/articles PMC 404759. Like our GPT, try our full AI-powered search engine and academic features for free at consensus.consensus.app/chatgpt. Critique of the article. Title: Impact of Plasmodium falciparum co-infection on longitudinal Epstein-Barr virus kinetics in Kenyan children. Strengths. 1. Significant contribution. The study provides valuable insights into the interaction between malaria and EBV, elucidating the potential mechanisms contributing to endemic Burkitt lymphoma. 2. Robust methodology. The use of quantitative polymerase chain reaction qPCR and detailed statistical models to analyze EBV kinetics is commendable. 3. Longitudinal data. The study benefits from a longitudinal cohort which allows for a detailed analysis of infection dynamics over time. Weaknesses. 1. Limited scope. The study focuses exclusively on children from 2 regions in Kenya, which may limit the generalizability of the findings to other populations or geographic areas with different malaria transmission intensities. Number 2, data gaps. There are periods with missing data, which could affect the accuracy of the viral load measurements and the interpretation of the results. 3, lack of mechanistic insights. While the study identifies correlations between malaria and EBV dynamics, it provides limited mechanistic insights into how malaria exposure specifically alters EBV kinetics. 4. Simplistic modeling approach. The modeling approach, though useful, might be overly simplistic. More advanced modeling techniques, such as machine learning or deep learning, could provide deeper insights and more accurate predictions. Suggestions for the introduction discussion section. Introduction. 1. Global context. Expand the introduction to include a discussion of the global prevalence and impact of EBV and malaria, not just in Africa, but in other endemic regions. 2. Previous research. Provide a more detailed review of previous studies that have explored the interaction between malaria and other viral infections to contextualize the novelty of the current study. 3. Mechanistic hypotheses. Introduce potential mechanistic hypotheses on how Plasmodium falciparum might affect EBV infection dynamics, drawing on immunological and virological theories. Discussion. 1. Implications for public health. Elaborate on the public health implications of the findings, particularly regarding malaria control measures and their potential to reduce the burden of EBV-associated diseases. 2. Therapeutic interventions. Discuss the potential for therapeutic interventions that target both malaria and EBV and how such integrated approaches might mitigate the risk of Burkitt lymphoma. 3. Limitations and future directions. provide a more detailed discussion of the study's limitations, including the geographic and demographic constraints, and outline specific future research directions. Suggest studies that use more advanced analytical techniques or that investigate the immune response in more detail. 4. Comparative analysis. Include a comparative analysis of the findings with other regions where malaria is prevalent, but the incidence of Burkitt lymphoma is different to highlight any unique regional factors. 5. Policy implications. Suggest policy changes or recommendations for health authorities in malaria endemic regions, emphasizing the need for comprehensive approaches to managing co-infections. Additional points for discussion. Genetic factors. Consideration of genetic factors that may influence susceptibility to both malaria and EBV, which could help in understanding individual differences in disease outcomes. Environmental and socioeconomic factors. Discussion on how environmental and socioeconomic factors might influence the observed patterns of infection and disease, providing a more holistic view of the epidemiological landscape. Integration with other diseases. Explore the interactions between malaria, EBV, and other prevalent diseases in the region, such as HIV, to provide a more comprehensive picture of the health challenges faced by these populations. By addressing these points, the article can provide a more thorough and impactful contribution to the understanding of malaria and EBV interactions and their implications for public health. For more detailed insights, refer to the full study here, ncpi.ncbi.nlm.nih.gov/pmc articles/C4SOR75/. Like our GPT, try our full AI-powered search engine and academic features for free at consensus.consensus.app/UTM source ChatGPT. Alternative explanations or interpretations of the results. 1. Genetic susceptibility. Genetic differences among Kenyan children studied might influence their susceptibility to both Plasmodium falciparum and Epstein-Barr virus (EBV) infections. Variations in immune response genes could explain the observed differences in EBV load and reactivation frequency. 2. Environmental factors. Factors such as nutritional status, access to healthcare, and exposure to other infections or environmental toxins might also play a role in the differences observed between high and low malaria transmission rates. These factors could affect immune function and susceptibility to infections. 3. Co-infections with other pathogens. The presence of other co-infections, e.g., other parasitic, bacterial, or viral infections could modulate the immune response and affect EBV dynamics. The interactions between multiple pathogens could lead to complex immune responses that influence EBV load and reactivation. 4. Immune system modulation by malaria. Malaria-induced immune modulation might not only directly affect EBV kinetics, but could also lead to long-term changes in immune function. These changes might alter the host's ability to control EBV, leading to higher viral loads and more frequent reactivations. 5. Behavioral and socioeconomic differences. Behavioral and socioeconomic differences between the populations in high and low malaria transmission areas might contribute to the observed results. Differences in health-seeking behavior, exposure to infections, and overall health status could influence EBV dynamics. Follow-up research project. Investigating the genetic and immunological mechanisms underlying Plasmodium falciparum and Epstein-Barr virus co-infection in Kenyan children. Objective: to identify the genetic and immunological factors that influence the dynamics of EBV infection in the context of Plasmodium falciparum co-infection, and to understand the mechanisms by which malaria exposure affects EBV load and reactivation. Study design: 1. Cohort selection. Recruit a new cohort of children from high and low malaria transmission regions in Kenya. Include a larger sample size to enhance the statistical power of the study. 2. Genetic analysis. Collect blood samples for genetic analysis to identify polymorphisms in immune response genes, e.g., HLA cytokine genes that may influence susceptibility to malaria and EBV. Perform genome-wide association studies, GWAS, to identify genetic variants associated with differences in EBV load and reactivation frequency. 3. Immunological profiling. Conduct detailed immunological profiling of the children, including flow cytometry to analyze immune cell subsets and cytokine production. Assess the function of EBV-specific and malaria-specific immune responses, including T-cell and B-cell responses. 4. Longitudinal monitoring. Monitor EBV and Plasmodium falciparum infection dynamics longitudinally using quantitative polymerase chain reaction, qPCR, to measure viral and parasitic loads. Track clinical outcomes and health status, including incidence of Burkitt lymphoma and other infections. 5. Environmental and behavioral factors. Collect data on environmental and socioeconomic factors, including nutrition, access to healthcare, and exposure to other infections. Use this data to control for potential confounding factors in the analysis. Data analysis. Multivariate analysis. Use multivariate statistical models to analyze the impact of genetic, immunological, environmental, and behavioral factors on EBV dynamics. Machine learning. Employ machine learning techniques to identify patterns and predictors of EBV reactivation and high viral loads. Expected outcomes. Identification of genetic variants and immune mechanisms that influence EBV infection dynamics in the context of malaria. Insights into how malaria-induced immune modulation affects long-term EBV control. Development of targeted interventions to reduce the risk of EBV-related diseases, such as Burkitt lymphoma in malaria-endemic regions. This follow-up study aims to provide a comprehensive understanding of the complex interplay between malaria and EBV, potentially leading to novel strategies for preventing and managing co-infections and their associated complications. For more detailed insights, refer to the full study here, ncb.ncbi.nlm.nih/articles/PMC4047-. Like our GPT, try our full AI-powered search engine and academic features for free at consensus.app, consensus.app, UTM source equals ChatGPT. Hashed impact of Plasmodium falciparum co-infection on longitudinal Epstein-Barr virus kinetics in Kenyan children. Table of contents. 1. Introduction. 2. Related research. 3. Research methodology. 4. Results. 5. Discussion and conclusion. 6. References. 1. Introduction. Epstein-Barr virus, EBV, is a ubiquitous human herpes virus that persists in the body after primary infection, typically in a latent state within B lymphocytes. Plasmodium falciparum, the parasite responsible for the most severe form of malaria, is endemic in many parts of Africa and contributes to significant morbidity and mortality. Co-infection with EBV and P. falciparum has been linked to the development of endemic Burkitt lymphoma, BL, the most common pediatric cancer in equatorial Africa. This study investigates the impact of P. falciparum co-infection on the longitudinal kinetics of EBV in Kenyan children, aiming to elucidate the mechanisms by which malaria influences EBV dynamics and contributes to BL pathogenesis. Hermer 2. Related research. Previous studies have established a connection between malaria and increased EBV reactivation and viral load. It is known that P. falciparum infection can induce polyclonal B-cell activation, which may facilitate EBV reactivation. Additionally, regions with high malaria transmission tend to exhibit higher rates of EBV-associated cancers. The intricate relationship between these two infections suggests that malaria might play a critical role in modulating the host's immune response to EBV, thereby affecting its persistence and reactivation cycles. Cohort selection. 2 cohorts of infants from Kisumu, high malaria transmission, and Nandi, low malaria transmission, in Kenya were monitored from birth up to 7 years of age. Data collection. Blood samples were collected approximately monthly during the first year and every 4 to 10 months thereafter. Quantitative polymerase chain reaction, qPCR, was used to measure EBV and P. falciparum DNA in whole blood. Statistical analysis. Statistical models, including Cox proportional hazards regression and Poisson regression, were employed to analyze the relationship between malaria exposure and EBV dynamics. Survival analysis was conducted to compare the time to EBV detection between the high and low malaria transmission regions. 4 results. 1. Early EBV infection and higher viral load. Children in high malaria transmission areas exhibited earlier EBV infections and higher viral loads during primary infection compared to those in low transmission areas. 2. Prolonged EBV DNA detection. The duration of detectable EBV DNA episodes was longer in children from high malaria transmission areas, with shorter intervals between reactivation episodes. 3. Concurrent P. falciparum infection. The presence of P. falciparum infection significantly increased the likelihood of EBV reactivation, indicating that malaria acts as a trigger for EBV replication. 5. Discussion and conclusion. The study's findings indicate that P. falciparum co-infection significantly affects EBV infection dynamics, leading to higher viral loads and more frequent reactivation episodes. This interaction likely contributes to the increased risk of developing Burkitt lymphoma in regions with high malaria transmission. The results suggest that controlling malaria could potentially reduce the incidence of EBV-related cancers by limiting viral reactivation. Alternative explanations: genetic susceptibility and environmental factors might also influence the observed differences in EBV dynamics. The role of other co-infections and overall health status could further modulate immune responses and EBV behavior. Follow-up research. Future studies should explore the genetic and immunological mechanisms underlying the observed interactions. A prospective cohort study incorporating advanced techniques like deep learning for predictive modeling could provide deeper insights into the factors influencing EBV kinetics and the development of effective intervention strategies. 6 references: Rinaldi A., Schlub T., Chelimo K. et al., 2016. Impact of Plasmodium falciparum co-infection on longitudinal Epstein-Barr virus kinetics in Kenyan children. Journal of Infectious Diseases, 236, 985-991. Link htabsacademic.oup.com/I2136 985/24593. PLOS pathogens. A molecular link between malaria and Epstein-Barr virus reactivation. Link at journals.plos.org/PLOS pathogen/article .1371/federaljournal.0049. Mormon AM et al. Impact of malaria on shaping immunity to EBV and the etiology of Burkitt lymphoma. Grantome. /grantome.com/grant01 CA 189806002. Summary with emphasized keywords: endemic Burkitt lymphoma, Epstein-Barr virus, EBV, Plasmodium falciparum, co-infection, higher rate of EBV expansion, primary infection, longer episodes of EBV DNA detection, shorter intervals, subsequent episodes, concurrent P. falciparum parasitemia, increased likelihood of EBV peaks, increased EBV growth, Burkitt lymphoma pathogenesis. For more detailed insights, refer to the full study here, academic.com/JADS article 213/6/45962. Summary with emphasized keywords: endemic Burkitt lymphoma, EBV, and Plasmodium falciparum co-infection. Modeling approach: study EBV infection kinetics, high malaria transmission, higher EBV expansion rate during primary infection, longer episodes of EBV DNA detection, shorter intervals between EBV reactivations, concurrent P. falciparum parasitemia, increased EBV growth, pathogenesis of endemic Burkitt lymphoma. This suggests a significant relationship between malaria and EBV dynamics, leading to increased Burkitt lymphoma risk in high malaria transmission areas. Summary with emphasized keywords: Burkitt lymphoma, discovered in 1958, most common childhood cancer in equatorial Africa. Risk factors: Epstein-Barr virus, EBV, and Plasmodium falciparum malaria. Seropositive for EBV, most African children by 3 years of age, some before 6 months. EBV persistence, low levels in adults in the UK and US, higher levels in Kenya and Gambia. Chronic repeated P. falciparum co-infection, EBV reactivation, increased latently infected B cells. Serological evidence, EBV reactivation in malaria holoendemic regions. Malaria transmission intensity influences age of primary EBV infection, contributes to higher EBV load. This emphasizes the interplay between malaria and EBV in increasing the risk of Burkitt lymphoma in children in endemic regions. Summary with emphasized keywords. Age of primary EBV infection, younger in regions of high malaria transmission, ongoing P. falciparum infection, and younger age predictors for higher total EBV load within first 24 months. Early EBV infection may program long-term lack of control, higher viral loads. High malaria transmission not sole factor, adults moving to these regions also at increased Burkitt lymphoma risk. Syndemic role of ongoing P. falciparum infections driving elevated EBV loads. Importance of understanding P. falciparum exposure mechanism on EBV load for further study. This highlights the significant role of malaria in influencing EBV dynamics and the need for further research on this interaction to understand its impact on Burkitt lymphoma. Summary with emphasized keywords. Cohort details, infants enrolled within 1 month of birth in Kisumu, high malaria transmission, and Nandi, low malaria transmission, Kenya. Monitoring, blood specimens collected monthly during first 12 months, then every 4 to 10 months up to 7 years. qPCR, used to measure EBV and P. falciparum DNA in whole blood specimens. Detection limit, 2 copies micrograms of human DNA. Antibodies, used to detect EBV load and determine age of primary EBV infection. Total EBV DNA includes latently infected B cells and EBV virions DNA fragments. Correlation, EBV DNA levels in plasma and cellular fractions significantly correlated. This emphasizes the rigorous methodology and detailed monitoring used in the study to understand the interplay between malaria and EBV infections in children. Summary with emphasized keywords. Ethical approval, approved by Kenya Medical Research Institute Ethical Review Committee, Kisumu. Institutional review boards approved by SUNY Upstate Medical University, Syracuse, New York, and University of Massachusetts Medical School, Worcester. This ensures the study adheres to ethical standards set by multiple research institutions. Summary with emphasized keywords. Comparison. Time to an event between groups. Children in regions of high and low malaria transmission. Log rank test used for significant differences. Modeling. Infection rates with exponential decay and constant rate. Delay included exponential decay with K, infection rate, and T on, time delay. Cox proportional hazard regression. Time to second EBV detection associated with age at first EBV detection, geographic location, Kisumu versus Nandi, P, falciparum parasite level, EBV load, Mann-Whitney U-test, compare single continuous variable between groups. Multivariable linear regression, effect of time to first EBV detection on viral growth rate. Poisson regression, relationship between number of viral reactivation events, age of EBV detection, malaria exposure, ecological variable. Software, analysis performed in R version 3.0.2. This outlines the comprehensive statistical approach used to analyze the impact of malaria on EBV infection dynamics, incorporating various regression models and statistical tests. Hadashat summary with emphasized keywords, initial infection with EBV, measured from birth to evidence of EBV infection, seroconversion, or EBV DNA detection. Earlier age of EBV infection in high malaria transmission areas. Force of infection, risk of infection per day, not significantly different between high and low transmission areas. Exponential survival curve, fitted with delay. Delay until infection, 4.6 months high transmission versus 5.9 months low transmission, p.001. Force of infection shared K 0.35 per month, median time to infection 2 months rise, p equals 0.13 t. Weibel with delay distribution, tested but did not fit significantly better. High transmission p equals 7.1, low transmission p equal 0.112. This summarizes the comparison of seroconversion rates and viral load detection between high and low malaria transmission areas, emphasizing the differences in timing and the statistical modeling approach used. Summary with emphasized keywords. Higher median EBV load observed in children from malaria endemic regions. Total EBV load compared using area under the curve AUC of viral load versus time. First EBV episode defined from first detection of EBV DNA until it becomes undetectable. Significant difference. Higher total EBV load in children from high malaria transmission regions during the first infection episode. Median AU, Terstat 8.98 versus 6.97 log 10 EBV copies per grams of DNA per month, p.001. This summarizes the comparison of EBV load during primary infection in infants, highlighting the significant differences in viral loads between children in high and low malaria transmission areas. Summary with emphasized keywords. Figure 2 overview. Time course of EB DNA detection shown for 4 subjects. High malaria transmission, Kisumu, Kenya. Higher total viral load during the first infection compared to low malaria transmission, Nandi, Kenya. Higher EBV growth rate observed in children from Kisumu during primary EBV DNA detection. Higher EBV expansion rate also seen during secondary EBV DNA detection in children from Kisumu. Measure of viral expansion, corrected for testing gap. This highlights the significant differences in EBV kinetics between children in high and low malaria transmission areas, emphasizing higher viral loads and growth rates in regions with higher malaria transmission. Summary with emphasized keywords. Higher total EBV load could be due to longer persistence of the first episode or higher initial growth rate. Growth rate measurement, typically between two closely spaced viral load measurements. Data issue, often failed to sample two successive measurements during the expansion phase. New method, estimating viral growth using the last negative and first positive reading. Incorporates duration of the testing gap. Ensures accurate estimates by using a narrow testing gap to avoid underestimation. This describes the methodology for accurately estimating the EBV growth rate, emphasizing the challenges and the newly developed approach to account for data gaps. Summary with emphasized keywords. Chi viral expansion, higher in high malaria transmission region, peak 0.051. Doubling time of EBV load, high malaria transmission 1.6 days, low malaria transmission 2.1 days. Previous estimation, EBV doubling time 2.7 days during reactivation post-hematopoietic stem cell transplantation. Duration of EBV DNA detection, high malaria transmission, that's 6.3 months. Low malaria transmission, 4.9 months, p equal 0.042. Higher AUC of EBV DNA load due to both higher EBV growth rate and prolonged detection of EBV DNA. This summarizes the differences in EBV kinetics between high and low malaria transmission areas, highlighting the faster growth rate and prolonged DNA detection in regions with higher malaria transmission. Summary with emphasized keywords, EBV dynamics, higher expansion rate and AUC of EBV load with high malaria exposure. Multivariable linear regression used to study EBV expansion rate and AUC of EBV load. Malaria exposure, strong determinant of early EBV expansion rate, p equal 0.05, and major predictor of total EBV load in first episode, p equals 0.01. Age at first EBV detection, not a significant predictor of higher EBV expansion rate or AUC in first episode. Contradiction. Previous findings associated younger age of infection with higher total viral load over time. Current study focused on total AUC of EBV DNA load in the first episode, not affected by age at first detection. Frequency of subsequent episodes, likely affected by age at first EBV detection. This highlights the key findings that malaria exposure, rather than age at seroconversion, predicts EBV dynamics during primary infection, explaining differences in earlier studies and emphasizing the role of malaria in influencing viral kinetics. Summary with emphasized keywords. Table 1, predictors of the Epstein-Barr virus EBV DNA expansion rate per month during the first infection. Age at first EBV detection, all testing gaps, coefficient 0.01, p equals 0.74, not significant. Narrow testing gap, coefficient 0.16, p equals 1, not significant. Area of high versus low malaria transmission, all testing gaps, coefficient Franchra 1.16, 0.59 to 1.73, p equal to 1, significant. Narrow testing gap, coefficient 1.43, 0.7 to 2 to 2.13, p equal 0.05, significant. Testing gap, all testing gaps, coefficient 0.83, p minus 1.1 to 0.66, p 0.001, significant. Narrow testing gap, coefficient 0.03, 0.43, p equals 0.87, not significant. This table indicates that malaria transmission area significantly predicts the EBV expansion rate, while the age at first EBV detection is not a significant predictor. The testing gap is significant in the overall data, but not in the narrow testing gap subset. Summary with emphasized keywords. Table 2, predictors of the total Epstein-Barr virus EBV load during the first infection. Area of high versus low malaria transmission, coefficient 1.38, 0.57 to 2.19, p 0.01, significant. Age at first EBV detection, Mo, coefficient minus 0.6, minus 0.22 to 0.09, p equals 0.42, not significant. Testing gap, Mo, coefficient more 0.06, minor 0.18 to 0.31, p equals 0.61, not significant. The GER EBV load was calculated as the area under the viral load curve, AUC. The analysis included all patients with no restriction on the testing gap. This table indicates that the area of malaria transmission is a significant predictor of the total EBV load, while the age at first EBV detection and the testing gap are not significant predictors. Summary with emphasized keywords. Primary EBV infection. In immunocompetent adults with infectious mononucleosis, EBV load declines to undetectable levels within 8 to 15 months. Kenyan infant cohort, dynamic pattern of EBV DNA detection with subsequent episodes after initial infection. Episode of EBV DNA detection defined as continuous EBV DNA detection, separated by undetectable periods. High malaria transmission, children more likely to have subsequent EBV DNA detection episodes, 84% versus 76 strand P form. Survival curve analysis, shorter time to second EBV DNA detection in high malaria transmission region, P.001. Rate of EBV DNA detection, higher in high malaria transmission area, 0.16 versus 0.09 detections per month. Comparison to primary infection. Rate of detection during primary infection higher, 0.35 per month, indicating start development of immune control. This highlights the significant impact of malaria exposure on the timing and frequency of subsequent EBV reactivation episodes, with a shorter time to reactivation and higher detection rates in high malaria transmission regions. Summary with emphasized keywords. Figure 1, EBV infection timing, time to infection, high malaria transmission, Kisumu versus low malaria transmission, Nandi. Exponential survival curve, different starting points, same infection rate. Time to second EBV DNA detection from end of primary episode to start of second episode. Exponential model, more rapid onset of second EBV episode in Kisumu cohort. This highlights the differences in the timing of EBV infections and subsequent reactivations between children in high and low malaria transmission areas, emphasizing the rapid reactivation in high transmission regions. Summary with emphasized keywords. Factors for rapid second EBV episode, Cox proportional hazard regression with multiple covariates. Younger age at primary EBV infection, increased risk of second episode, HR 0.9 and 7, kind of during first infection. Higher viral load during first of second episode, 3 P 0.048. This indicates that while younger age at initial infection increases the likelihood of subsequent EBV reactivation, a higher initial viral load may induce better immunity, thus reducing the risk of a second episode. Summary with emphasized keywords. Table 3, predictors of the time to the second episode of Epstein-Barr virus EBV detection. Age at first EBV detection, HR 0.89, 0.83 to 0.97, P equals 0.007 significant. Total viral load AUC at first infection, HR 0.83, 0.76 to 0.91, P equals 0.448 significant. Area of high versus low malaria transmission, HR 2.26, 1.47 to 3.48, P 0.001 significant. There was no violation of the proportionate hazard assumption. This table shows that younger age at first EBV detection and higher malaria transmission significantly predict a shorter time to the second episode of EBV detection, while a higher initial viral load is associated with a reduced risk of a second episode. Summary with emphasized keywords. Multiple peaks in EBV DNA detection observed in many patients. Poisson regression used to analyze age at first EBV infection, malaria endemicity. Findings, younger age at initial EBV detection, more episodes of EBV DNA detection, P 0.031. Higher malaria exposure, more episodes of EBV DNA detection, P threats. This indicates that both younger age at initial EBV infection and higher malaria exposure are associated with increased frequency of EBV reactivation episodes. Summary with emphasized keywords. EBV growth rate analysis during secondary EBV DNA detection. Total EBV load calculated as AUC. Narrow testing gap used for accurate estimates due to increased testing gap with age. Higher distribution of Chi. Higher EBV growth rate for children from high malaria transmission, P.01. Mean doubling time. High malaria transmission, 2.2 days. Low malaria transmission, 3.17 days. Primary episode comparison, slower in secondary episode, 1.6 days versus 2.05 days. Significant predictor, malaria exposure for both EBV expansion rate and total EBV load. Duration of second episode, high malaria transmission, 6 months. Low malaria transmission, 4.6 months, P.009. This highlights that children in high malaria transmission regions have higher EBV growth rates and longer episodes of EBV DNA detection during secondary infection, with malaria exposure being a significant predictor. Summary with emphasized keywords. Role of malaria, previously considered as an ecological variable, high or low malaria transmission areas. Concurrent P. falciparum infection, presence of infection at any given time may influence EBV dynamics. Investigation, links between P. falciparum parasitemia levels detected by PCR and EBV DNA detection. This indicates that beyond just living in high or low malaria transmission areas, the actual presence of P. falciparum infection at the time of sampling can affect the dynamics of EBV infection in individuals. Summary with emphasized keywords. Tested influence. Time to first detection of EBV DNA by P. falciparum parasitemia level. Cox regression used with binary group variable, high-low malaria transmission, and P. falciparum DNA level as a time-dependent covariate. Findings. Presence of P. falciparum at sampling time point increases risk of first detection of EBV DNA, HR 1.84, CI 1.4 to 2.3, P equals 0.2. Independent of malaria exposure region, HR 2.95%, CI 2.1 P.001. This highlights that the presence of P. falciparum infection significantly increases the risk of first EBV DNA detection, independent of the overall malaria transmission level in the region. Summary with emphasized keywords. Modified Cox regression included P. falciparum parasitemia level as a time-dependent covariate for second EBV episode. Violation of constant HR assumption. Malaria region binary variable, P equals 0.0148. Log transform P. falciparum load, P.032. Stratified model, data split into second EBV DNA detection in 10 months after primary infection. Detection in 10 months after primary infection. Findings. Similar trends for age at first EBV infection, total EBV load during first episode, and malaria exposure group. Concurrent P. falciparum parasitemia, higher risk of second EBV DNA detection at any age. HR 1.53 for detection 10 months after primary infection, p equals 0.02. HR 1.24 for detection 10 months after primary infection, p equal 0.03. Conclusion, P. falciparum induces reactivation of latent EBV. This analysis confirms that P. falciparum parasitemia significantly increases the risk of second EBV DNA detection, supporting the hypothesis that malaria triggers EBV reactivation. Summary with emphasized keywords. Table 4 predictors, including Plasmodium falciparum level of the time to the second episode of Epstein-Barr virus EBV detection. 10 months from the end of primary EBV detection, P. falciparum level, HR 1.53, 1.23 to 1.91, P equals 0.002. Total viral load, AUC at first infection, HR 0.5 to 0.87, P equals 0.009. Area of high versus low malaria transmission, HR 2.92, 1.98 to 4.3, P equals 0.014. Age at first infection, HR 0.61, 0.42 to 0.89, P equals 0.01. TR 10 months from the end of primary EBV detection, P. falciparum level, HR 1.24, 1.12 to 1.37. Total viral load, AUC at first infection, HR 0.53, 0.37 to 0.76, P 0.001. Area of high versus low malaria transmission, HR 1.87, 1.15 to 3.01. Age at first infection, HR 0.66 to 0.98, P 9 days. Note, time of risk divided into those 10 months and a due 10 months post-primary EBV infection to uphold proportion hazard assumption. This table shows that P. falciparum level, total viral load, and had area of malaria transmission significantly influenced the time to second EBV detection with different impacts within 10 months and 10 months intervals after primary infection. Summary with emphasized keywords, EBV and P. falciparum co-infection, risk factor for endemic Burkitt lymphoma. Key determinants, degree of malaria exposure, age at primary EBV infection. Findings, malaria exposure, key determinant of primary EBV infection kinetics, independent of age. Higher EBV expansion rate and longer duration of detectable EBV DNA in high malaria transmission areas. Higher total EBV load in high malaria transmission regions during primary and subsequent episodes. This highlights that malaria exposure plays a crucial role in influencing EBV dynamics, contributing to higher viral loads and prolonged detection in areas with high malaria transmission, which is significant for understanding the interaction between these infections and their role in Burkitt lymphoma development. Hadish's summary with emphasized keywords. Figure 3, role of malaria exposure and age of primary EBV infection. High malaria exposure leads to higher rate of EBV expansion, higher total viral load, AUC, longer durations of detectable EBV DNA during primary and subsequent episodes. Plasmodium falciparum parasitemia increases risk of first and second EBV infection. Early age at primary EBV infection, independently associated with shorter time to the second episode of EBV DNA detection. This schematic illustrates that malaria exposure significantly impacts EBV dynamics, resulting in higher viral loads and prolonged detection periods, with P. falciparum playing a critical role in increasing the risk of multiple EBV infections. Summary with emphasized keywords. Factors affecting subsequent EBV peaks. Earlier age at EBV infection. Residence in high malaria transmission regions. Both decreased time between first and second EBV load peaks. Increased frequency of second peak in EBV load. Infant EBV oscillations different from acute infectious mononucleosis in young adults. In AM, EBV load resolves over 1 year to a steady state level. Elevated EBV load linked to reactivation in immunosuppressed patients. Study limitations cannot distinguish between reactivation of primary infection and true secondary infection, superinfection. Slower EBV expansion rate during secondary detection compared to primary infection. Further research needed. Identify molecular and cellular factors promoting high EBV levels in malaria-exposed individuals. Study immune responses to EBV during primary and subsequent episodes. This highlights the impact of early age and high malaria exposure on EBV dynamics, emphasizing the need for further research to understand the mechanisms behind EBV reactivation and persistence in these conditions. Summary with emphasized keywords. Concurrent P. falciparum infection. Increased risk for first EBV detection. Increased risk for second EBV detection. This underscores the significant impact of concurrent malaria infection on the likelihood of detecting EBV, highlighting the interplay between these two infections. Summary with emphasized keywords. Proposed model, increased susceptibility of B cells to EBV infection. Combined factors, increased mutation, reduced apoptosis. Result, drives oncogenesis as a risk for Burkitt lymphoma in malaria endemic regions. Additional contribution of malaria expands pool of latently infected B cells. These B cells are susceptible to a second oncogenic hit. This model suggests that malaria may enhance the risk of Burkitt lymphoma by increasing the number of EBV-infected B cells prone to further oncogenic events. Summary of endemic Burkitt's lymphoma as a polymicrobial disease. New insights on the interaction between Plasmodium falciparum and Epstein-Barr virus. Introduction. Endemic Burkitt's lymphoma, EBL, is a childhood cancer prevalent in equatorial Africa, strongly associated with both Epstein-Barr virus, EBV, and Plasmodium falciparum malaria. This review explores the molecular interactions between these pathogens and their role in EBL pathogenesis. Key findings. 1. Relationship between EBV and malaria. EBV establishes a lifelong infection with virus shedding in saliva, and African children are commonly infected early. Malaria induces polyclonal B-cell activation, hypergammaglobulinemia, and increases circulating EBV levels. 2. Molecular link. Recent data suggest a link between Plasmodium falciparum's cysteine-rich interdomain region 1, CIDR1, and EBV. CIR1, part of the P, falciparum membrane protein PFEM1, activates B cells and increases EBV reactivation, especially in memory B cells where EBV persists. 3. EBV reactivation. CIDR1 directly induces EBV reactivation in EBV-infected B cells. This interaction leads to increased viral loads and B cell activation, enhancing the risk of EBL development. 4. Polymicrobial disease pathogenesis. The concurrent infection with P, falciparum, and EBV leads to an environment conducive to EBL. Chronic immune activation and viral reactivation caused by malaria heighten the risk of lymphoma by promoting genetic mutations and prolonged B-cell survival. Conclusion. The interaction between Plasmodium falciparum and Epstein-Barr virus underpins the polymicrobial nature of endemic Burkitt's lymphoma. Understanding these mechanisms offers insights into disease pathogenesis and potential therapeutic targets. Citations: Chain et al., 2000, consensus.app/papers/endemic Burkitt's lymphoma disease insights interaction Chain, 90F665145 ECA111F2BB4F, UTM Circles ChatGPT, Rochford, Cannon, and Mormon 5. Consensus.app/papers/endemic Burkitt's lymphoma disease-Rochford/CD589266759EABB421D/UTM Swassen Chat Papers/link Plasmodium falciparum malaria and Burkitt Thorley Lawson 4 DFNO5 B84B2/ in NIEC2 9424B ChatGT. Consensus.app/papers/TLR9 is triggering Burkitt's lymphoma cell lines, suppresses Zoner/43D7156695091A435B1BF10/ likes our GP. Like our GP, try our full AI-powered search engine and academic features for free at consensus/consensus.app/UTSource=ChatGPT. Summary in one sentence. The study explores the molecular interactions between Plasmodium falciparum and Epstein-Barr virus, EBV, in the pathogenesis of endemic Burkitt's lymphoma, EBL, demonstrating how P. falciparum antigens can induce EBV reactivation and increase the risk of EBL in children in malaria-endemic regions. Purpose and necessity of the study. Purpose. 1. To investigate the molecular mechanisms linking Plasmodium falciparum malaria and Epstein-Barr virus in the development of endemic Burkitt's lymphoma. 2. To identify specific interactions between P. falciparum antigens and EBV that contribute to increased viral reactivation and lymphoma risk. 3. To provide new insights into the concept of polymicrobial disease pathogenesis in EBL. Necessity. 1. Endemic Burkitt's lymphoma is a significant health issue in equatorial Africa, being the most common childhood cancer in these regions. 2. Understanding the interactions between P. falciparum and EBV could lead to better prevention and treatment strategies for EBL. 3. The study aims to fill the research gap regarding the combined impact of malaria and EBV on the immune system and lymphoma development. Subject, method, and results. The study focuses on children in malaria-endemic areas who are at high risk for endemic Burkitt's lymphoma due to the co-infection of Plasmodium falciparum and Epstein-Barr virus. Method: the research involved analyzing the interaction between the CIDR1 high region of P. falciparum's PFEM1 protein and EBV in B cells. Various laboratory techniques, including real-time PCR and cell culture assays, were used to study EBV reactivation and B-cell activation in response to CIDR1-HA. Results. CIDR1A was identified as a polyclonal B cell activator that increases B cell survival and preferentially activates memory B cells, leading to EBV reactivation. The study demonstrated that EBV reactivation can be directly induced by P. falciparum antigens, resulting in increased viral loads and heightened risk of EBL development in malaria endemic areas. The findings support the concept of EBL as a polymicrobial disease, with both EBV and P. falciparum playing crucial roles in its pathogenesis. Like our GPT, try our full AI-powered search engine and academic features for free at consensus.app and consensus source equals ChatGPT. Conclusion. Implications and follow-up research plans. Implications. 1. The study establishes a direct molecular link between Plasmodium falciparum malaria and Epstein-Barr virus reactivation, suggesting that chronic malaria infection contributes significantly to the development of endemic Burkitt's lymphoma. 2. The findings support the concept of endemic Burkitt's lymphoma as a polymicrobial disease, highlighting the critical role of malaria-induced immune modulation and EBV reactivation in lymphoma pathogenesis. 3. The research underscores the need for targeted interventions that address both malaria and EBV infections to reduce the incidence of EBL in malaria-endemic regions. Follow-up research plans suggested by researchers. 1. Further investigation into the specific molecular mechanisms by which CIDR1 high induces EBV reactivation in B cells. 2. Studies to explore the potential for developing therapeutic strategies that inhibit CIDR1-induced EBV reactivation. 3. Longitudinal studies to assess the impact of anti-malarial treatment on EBV load and the subsequent risk of developing EBL. 3 additional follow-up research directions. 1. Immunological studies. Examine the broader immune response to P. falciparum infection and its impact on other latent viral infections beyond EBV to understand the full scope of polymicrobial interactions in endemic regions. 2. Epidemiological research. Conduct large-scale population studies to quantify the incidence of EBL in relation to malaria transmission intensity and EBV seroprevalence, providing more detailed data on risk factors and regional variations. 3. Vaccine development. Explore the potential for vaccines targeting both P. falciparum and EBV to reduce the burden of EBL, including trials of candidate vaccines and assessment of their efficacy in preventing EBV reactivation and lymphoma development in endemic areas. Like our GPT, try our full AI-powered search engine and academic features for free at consensus.app/UTM-source-chat-GPT. Novelty, most important finding. The most important finding of this paper is the demonstration that the Plasmodium falciparum antigen CIDR1-ANHA can directly induce Epstein-Barr virus EBV reactivation in B cells, which significantly enhances the risk of developing endemic Burkitt's lymphoma, EBL, in children living in malaria endemic areas. This is the first study to show a direct molecular interaction between a malaria pathogen and EBV, establishing a clear link in the polymicrobial pathogenesis of EBL. Keywords: 1. Endemic Burkitt's lymphoma, 2. Plasmodium falciparum, 3. Epstein-Barr virus, EBV, 4. CIDR1A, 5. Polymicrobial disease, 6. Malaria, 7. B-cell activation, 8. Viral reactivation, 9. Immune modulation, 10. Childhood cancer. Like our GPT, try our full AI-powered search engine and academic features for free at consensus.app/UTM-source-chat-GPT. Concept comparison. Concept endemic Burkitt's lymphoma, EBL. Feature endemic Burkitt's lymphoma, EBL. Sporadic Burkitt's lymphoma, SBL. Associated pathogens, Epstein-Barr virus. Geographic distribution, Equatorial Africa, Papua New Guinea. Worldwide, particularly in developed countries. Associated pathogens, Epstein-Barr virus, EBV, and Plasmodium falciparum. EBV, less frequently, no association with malaria. Age group, children, peak incidence at 5 to 8 years. Children and young adults. Immunological context, high EBV seroprevalence, chronic malaria infection, normal or slightly elevated EBV seroprevalence. Pathogenesis, polymicrobial interaction involving immune modulation by malaria. Genetic mutations, EBV infection, but no clear link with malaria. Incidence rate, high in malaria endemic regions, lower and more sporadic. Rationale of using real-time PCR method. Real-time PCR, polymerase chain reaction, is used in this study to quantify EBV DNA levels due to its high sensitivity and specificity. This method allows for 1. Accurate quantification of viral load in biological samples, even at low concentrations. 2. Monitoring of changes in EBV DNA levels over time, which is crucial for understanding the dynamics of viral reactivation. 3. Rapid and reliable detection, making it suitable for large-scale studies and clinical applications. Questions and answers to assess comprehension. 1. Q. What is the primary focus of the study? The primary focus is on the interaction between Plasmodium falciparum and Epstein-Barr virus in the pathogenesis of endemic Burkitt's lymphoma. 2. How does CIDR1-Fa of Plasmodium falciparum contribute to EBV reactivation? CIDR1A acts as a polyclonal B-cell activator, preferentially activating memory B cells where EBV persists, leading to increased viral reactivation and load. 3. Why is endemic Burkitt's lymphoma considered a polymicrobial disease? It is considered a polymicrobial disease because it involves interactions between multiple pathogens, specifically EBV and Plasmodium falciparum, which together contribute to its pathogenesis. 4. What methods were used to demonstrate the interaction between CIDR1-Han and EBV? The study used real-time PCR and cell culture assays to show that CIDR1A can induce EBV reactivation in B cells. 5. What implications do the study's findings have for preventing endemic Burkitt's lymphoma? The findings suggest that targeting both malaria and EBV infections through interventions could reduce the incidence of EBL in endemic regions. Like our GPT? Try our full AI-powered search engine and academic features for free at consensus.app/usource-chat-GPT. Recommended methodology, machine learning, deep learning. Method, deep learning for predictive modeling. Rationale, deep learning and machine learning techniques can be used to predict the likelihood of EBV reactivation and EBL development by analyzing large data sets of biological markers and clinical data. These methods can help identify complex patterns and interactions between multiple factors, such as genetic, immunological, and environmental variables, that traditional statistical methods might miss. Example techniques. 1. Convolutional neural networks, CNNs. Application. Analyzing high-throughput sequencing data to identify genetic mutations associated with increased risk of EBL. Title example. Deep learning approaches for genomic prediction of disease risk. 2. Recurrent neural networks, RNNs, or long short-term memory networks, LSTMs. Application. Predicting viral reactivation dynamics based on longitudinal patient data. Title example, predicting viral reactivation in endemic Burkitt's lymphoma patients using recurrent neural networks. 3. Random forest or gradient boosting machines. Application, identifying key risk factors and their interactions from clinical and environmental data. Machine learning-based identification of risk factors for endemic Burkitt's lymphoma. 4. Autoencoders. Application, reducing the dimensionality of large datasets and identifying underlying patterns that contribute to disease development. Title example, dimensionality reduction in high-dimensional genomic data using autoencoders for disease prediction. Example study title, using machine learning, utilizing machine learning to predict Epstein-Barr virus reactivation and endemic Burkitt's lymphoma risk in malaria endemic regions. Potential benefits of using deep learning in this context. 1. Enhanced predictive accuracy. Deep learning models can handle large and complex datasets, potentially improving the accuracy of predictions regarding EBV reactivation and EBL risk. 2. Identification of nonlinear interactions. These models can uncover nonlinear interactions between multiple risk factors, which are often missed by traditional analysis methods. 3. Automation and scalability. Once trained, deep learning models can automatically analyze new data, making them scalable for large population studies. Previous study example. While specific titles may vary, previous studies have used machine learning for similar purposes, such as predictive modeling of viral reactivation using machine learning techniques, deep learning models for predicting disease outcomes in epidemiological studies. These studies demonstrate the feasibility and effectiveness of applying machine learning and deep learning techniques to predict disease risk and understand pathogen interactions in complex biological contexts. Shesh article critique as a strict reviewer. Title, Endemic Burkitt's lymphoma as a polymicrobial disease. New insights on the interaction between Plasmodium falciparum and Epstein-Barr virus. 1. Abstract. Strengths. The abstract provides a comprehensive overview of the study, summarizing the key findings and the novel interactions discovered. Weaknesses. The abstract could be more concise. It includes some detailed information that might be better suited for the main text. 2. Introduction. Strengths. The introduction effectively sets the context for the study by discussing the known associations between EBV, malaria, and EBL. Weaknesses. The introduction lacks a clear statement of the hypothesis. Additionally, it could better highlight the gaps in existing research that this study aims to fill. Methods. Strengths. The methods are detailed and appropriate for the study's objectives, providing clear descriptions of the experimental procedures. Weaknesses. The study could benefit from a more robust statistical analysis section detailing the specific statistical tests used and their justification. Additionally, the sample size and selection criteria for B cells and patient samples should be more explicitly discussed to address potential biases. 4. Results. Strengths. The results are clearly presented with relevant figures and tables that help illustrate the findings. Weaknesses. There is a lack of clarity in distinguishing between statistically significant and non-significant results. The results section could benefit from a more in-depth analysis and interpretation of the data. 5. Discussion. Strengths. The discussion effectively contextualizes the findings within the broader literature and suggests plausible mechanisms for the observed interactions. Weaknesses. The discussion sometimes speculates without sufficient backing from the data. More emphasis should be placed on the limitations of the study, including potential confounders and biases. The discussion should also provide a clearer outline of future research directions based on the study's findings. 6. Conclusion strengths. The conclusion succinctly summarizes the study's contributions and implications. Weaknesses. It could be strengthened by reiterating the study's main limitations and proposing specific, actionable steps for future research. 7. References. Strengths. The references are comprehensive and relevant to the study. Weaknesses. Ensure that all citations are up to date and include the most recent and relevant studies in the field. Additional points for introduction discussion. Introduction. 1. Historical context. Provide more background on the discovery of endemic Burkitt's lymphoma and its initial associations with EBV and malaria. 2. Research gaps. Clearly outline the specific research gaps this study aims to address, such as the lack of detailed understanding of the molecular mechanisms linking P. falciparum and EBV. 3. Hypothesis. Explicitly state the hypothesis of the study to guide the reader on what the research is testing. Discussion. 1. Comparative analysis. Include a comparison of the findings with other studies that have examined the interaction between EBV and malaria, highlighting consistencies and discrepancies. 2. Mechanistic insights. Provide deeper insights into the potential molecular mechanisms underlying the interaction between CIDR1 and EBV reactivation. 3. Public health implications. Discuss the public health implications of the findings, particularly in terms of developing integrated strategies for managing malaria and preventing EBL. 4. Limitations. Acknowledge and discuss the limitations of the study in more detail, including the sample size, geographic limitations, and potential biases in data collection. 5. Future directions. Outline specific future research directions, such as investigating other P. falciparum antigens that might interact with EBV, or exploring the role of other co-infections in EBL pathogenesis. Conclusion. While this study provides valuable insights into the polymicrobial interactions contributing to endemic Burkitt's lymphoma, it could be strengthened by addressing the above critiques and expanding on certain sections. Incorporating these suggestions would enhance the robustness and impact of the research. Alternative explanations or interpretations of the results. 1. Host genetic factors. The interaction between Plasmodium falciparum and Epstein-Barr virus, EBV, might be influenced by specific host genetic factors that affect immune responses. Genetic polymorphisms in immune-related genes could modulate susceptibility to EBV reactivation and the development of endemic Burkitt's lymphoma, EBL. 2. Environmental and nutritional factors. Malnutrition and other environmental factors prevalent in malaria-endemic regions might also contribute to the observed increase in EBV reactivation and EBL risk. Poor nutrition could impair the immune system, making it more susceptible to chronic infections and reactivation of latent viruses. 3. Other co-infections. Besides malaria, other common infections in these regions might synergistically contribute to the observed immune modulation and increased EBV reactivation. For instance, infections with other parasites, bacteria, or viruses could further weaken immune control over EBV. 4. Immune evasion by EBV. EBV itself might possess mechanisms that enable it to exploit the immune modulation caused by malaria. For example, EBV could manipulate immune checkpoints or evade immune surveillance more effectively in the presence of malaria-induced immune changes. Follow-up research project. Title: Investigating the combined role of host genetics, environmentals, and co-infections in the pathogenesis of endemic Burkitt's lymphoma. Objectives: 1. To explore the genetic predispositions that may influence susceptibility to EBV reactivation and EBL development. 2. To assess the impact of environmental and nutritional factors on the immune system and their role in EBL pathogenesis. 3. To evaluate the contribution of other common co-infections in malaria endemic regions to EBV reactivation and EBL risk. Methodology. 1. Study design. Cohort selection. Enroll a large cohort of children from malaria endemic regions, including those with EBL, those with malaria but without EBL, and healthy controls. Data collection. Collect comprehensive data on genetic, environmental, nutritional, and infection-related variables. 2. Genetic analysis. Genome-wide association studies, GWAS. Conduct GWAS to identify genetic polymorphisms associated with increased susceptibility to EBV reactivation and EBL. Candidate gene approach. Focus on specific immune-related genes known to influence viral infections and lymphocyte function. 3. Environmental and nutritional assessment. Surveys and biomarkers. Use detailed surveys and biomarker analysis to assess nutritional status, environmental exposures, and socioeconomic factors. Statistical analysis. Analyze the correlation between these factors and the incidence of EBV reactivation and EBL. 4. Co-infection studies. Microbiome and virome analysis. Perform comprehensive analysis of the microbiome and virome in the cohort to identify other prevalent infections. Immunological profiling. Use flow cytometry and cytokine assays to profile immune responses in the presence of multiple infections. 5. Functional studies. In vitro experiments. Use in vitro models to study the interactions between P. falciparum antigens, EBV, and immune cells under different genetic and environmental conditions. Animal models. Develop animal models to validate the findings and explore mechanistic pathways. Expected outcomes. 1. Identification of genetic factors that predispose individuals to EBV reactivation and EBL. 2. A better understanding of how environmental and nutritional factors influence immune responses and EBL risk. 3. Insights into the role of other co-infections in modulating immune control over EBV. 4. Development of targeted intervention strategies that address genetic, environmental, and infectious risk factors for EBL. Significance. This follow-up research will provide a holistic understanding of the multifactorial nature of endemic Burkitt's lymphoma, potentially leading to more effective prevention and treatment strategies in malaria endemic regions. Like our GPT, try our full AI-powered search engine and academic features for free at consensus.app/consensus.app/UTM source equals ChatGPT. The polymicrobial nature of endemic Burkitt's lymphoma, interactions between Plasmodium falciparum and Epstein-Barr virus. Table of contents. 1. Introduction. 2. Related research. 3. Research methodology. 4. Results. 5. Discussion and conclusion. 6. References. 1. Introduction. Endemic Burkitt's lymphoma, EBL, is a highly aggressive B-cell lymphoma most commonly affecting children in malaria endemic regions of Equatorial Africa and Papua New Guinea. The polymicrobial nature of endemic Burkitt's lymphoma, interactions between Plasmodium falciparum and Epstein-Barr virus. Table of contents: 1. Introduction. 2. Related research. 3. Research methodology. 4. Results. 5. Discussion and conclusions. References. NAR 1. Introduction. Endemic Burkitt's lymphoma, EBL, is a highly aggressive B-cell lymphoma most commonly affecting children in malaria-endemic regions of Equatorial Africa and Papua New Guinea. Summary. Most prevalent childhood cancer in Equatorial Africa, malaria holoendemic areas. High-grade B-cell lymphoma, characterized by C-mic translocation and consistent presence of EBV. EBV infection, lifelong persistent infection, virus shedding into saliva, early infection in African children, seroconversion by age 3. Malaria's impact. Acute and chronic malaria profoundly affect B-cell compartment. Induces polyclonal activation, causes hypergammaglobulinemia, increases levels of circulating EBV. Interaction between pathogens. Very little research on direct interaction. Evidence suggests malaria enhances EBV reactivation. Key takeaways. Linked to both Plasmodium falciparum and EBV. Malaria infections increase risk of EBL through immune system modulation and EBV reactivation. Summary molecular link between Plasmodium falciparum, B-cell, and EBV, supports polymicrobial disease pathogenesis in EBL, CIDR1, identified as polyclonal B-cell activator, increases B-cell survival, preferentially activates memory B-cells where EBV persists. Mechanism, CIDR1 induces virus production in EBV-infected B-cell line Akata, also in latently infected primary B-cells from healthy carriers and children with EBL. Significance. First demonstration that EBV reactivation can be directly induced by another pathogen. Key takeaways. CIDR1 crucial in linking malaria and EBV reactivation. Polymicrobial pathogenesis key concept in EBL development. Summary, P. falciparum antigens, PMP1 can induce EBV reactivation during malaria infections. Increased viral load leads to polyclonal B-cell activation, enhances B-cell survival, risk of EBL, augmented risk of EBL development, particularly in children in malaria endemic areas. Key takeaways, critical in EBV reactivation. Increased viral load and B-cell activation elevate EBL risk in malaria endemic regions. Summary historical research, Dennis Burkitt, Ted Williams, Cliff Nelson, 1960s, 16,000 kilometers travel, 60 hospitals in East and Southern Africa. Findings: geographical distribution of sarcomas lymphoma correlated with malaria zones, temperature, rainfall, lymphoma linked to insect vectors, and malaria transmission. Malaria association: high incidence of lymphomas, 5 to 10 cases per 100,000 children in lowlands of Kenya, lower incidence in urban areas or highlands, found only in holo or hyperendemic areas for Plasmodium falciparum. Regions: Lymphoma Belt of Africa, Papua New Guinea, Irian Jaya in Asia. Key takeaways: Burkitt identified lymphoma malariae link in 1960s, high lymphoma incidence in malaria endemic areas. Lymphoma belt includes parts of Africa and Asia. Summary: Initial observation: Burkitt, 1957, Mulago Hospital, Kampala. Young boy with tumors in head and neck. Similar case in girl weeks later, fast-growing tumors, children died within weeks. Discovery: jaw tumors common in Uganda. Published observations in British Journal of Surgery, 1958. Named endemic Burkitt's lymphoma, EBL. Characteristics: aggressive B-cell lymphoma, C-mic translocation, associated with EBV and malaria. Also found in HIV-infected individuals. Tumor growth: high proliferative index, Ki67 95%, doubling time 24 to 26 hours. Tumor size can reach 10 to 15 centimeters in advanced cases. Key takeaways: Burkitt identified aggressive lymphoma in 1957, EBL linked to EBV, malaria, and C-mic translocation. Rapid tumor growth, high proliferative index. Summary: new data, 52 years after EBL description, emerging mechanisms of EV and P. falciparum interaction. Polymicrobial disease. EBL considered a polymicrobial disease. First human cancer associated with 2 interacting pathogens. Key takeaways: new insights into EBV and P. falciparum mechanisms. EBL as polymicrobial disease. First cancer linked to pathogen interaction. Summary: Plasmodium falciparum causes severe clinical malaria associated with EBL. Life cycle: Anopheles mosquito blood meal, parasite enters bloodstream, migrates to liver, infects and divides in hepatocytes. Hepatocyte bursts, releases merozoites into bloodstream. Merozoites invade erythrocytes, develop, and divide. 48-hour cycles can continue for up to 9 months. Immune system interaction: lives in bloodstream, constantly interacts with the immune system. Key takeaways: P. falciparum life cycle involves liver and erythrocytes. Parasites presence in bloodstream critical for understanding interaction with EBV. Malaria linked to EBL through immune system dynamics. Summary: Intraerythrocytic growth, proteins, PFEMP1 rifin, Steve or surfin, expressed and presented on human red blood cell RBC surface, PFEMP1. Adhesin enables infected RBCs to adhere in microvasculature, avoids clearance by spleen. Microvasculature, parasite thrives in hypoxic environment. Infected RBCs sequester, clogging microvasculature, leads to severe malaria. Immune evasion, antigenic variation of polypeptides, changes in receptor recognition and tissue tropism. Exposed variant surface antigen influences disease outcome. Achilles heel, host immune recognition of altered erythrocyte surface. Key takeaways. And other proteins crucial for RBC adhesion and immune evasion. Sequestration in microvasculature critical for malaria pathogenesis. Antigenic variation helps parasite evade immune response and affects disease severity. Summary: large protein, 200 to 350 kiloday, encoded by 60 VAR genes, single variant gene expressed per parasite at a time. Transcriptional silencing of other genes. Gene expression, same VAR gene expressed across generations. Switching rate, 1%. Immune evasion and prolonged infection. Gene structure, 2 exons conserved intron, Topstru-5A upstream flanking sequence. Common structural elements, N-terminal segment, Duffy binding-like domains, DBL, cysteine-rich interdomain regions, CI transmembrane lane, TM domain, C2 domain, acidic terminal segment, ATS. Function, related protein architectures. Example, VAR1 of FCRF1.2, parasite clone. Key takeaways. Crucial for immune evasion through antigenic variation. Encoded by multiple VAR genes with precise expression control. Common structural features across PFMP1 variants. Now for summary. Figure 1, molecular architecture of PMP1, encoded by VAR1 gene of FCR3S1.2 parasite, receptor specificities, heparan sulfate A31, PCAM1, CD36, receptors present on B cells. Key takeaways. PFEMP1 WARM binds to specific receptors on B cells. Receptors include HS, CD31, PCAM1, CD36. Summary. PMP1 NAS CIDR1A crystal structure, Malayan camp parasite strain, composed of 3 A helices connected by a loop to 3 additional helices. Structural relatedness, CIDR and DBL domains show structural similarity. PFEMP1 is a polymer of 3 helix bundles. Comparison. Similar structure to protein A of Staphylococcus aureus. Protein A, immunoglobulin binding polypeptide, B cell superantigen. Key takeaways. PFEMP1 CIDR1 structure involves 3 helix bundles. Structural similarity between CIDR, DBL domains, and protein A Summary: chronic malaria, common in highly endemic areas, numerous episodes in childhood. Clinical symptoms: not all infections result in clinical symptoms. Only a minority develop severe disease. Immunity development depends on intensity of transmission, function of age. Symptoms vary with transmission level. Key takeaways: high malaria exposure in endemic areas, immunity influenced by transmission intensity and age. Severe malaria affects a minority. Summary: mean parasite density and mean spleen size. Figure 2a, children in holoendemic malaria area. Parasite density and spleen size increase with age up to 6 years, then decline. Spleen size measurement. Hackett's method. Size 3, umbilical level to lower thoracic cage. Size 2, halfway to lower thoracic cage. Size 1, under thoracic cage. Clinical symptoms. Figure 2b, correlation with age, severe, mild, and asymptomatic phases. Severe mild symptoms peak around 2 to 4 years, transition to asymptomatic phase after 6 years. Key takeaways: parasite density and spleen size peak in early childhood. Spleen size measured by Hackett's method. Symptom severity decreases with age. Summary: clinical symptoms, rare with age in highly endemic malaria areas. Stepwise immunity development. Severe malaria first, then clinical symptoms. Slow immunity development. Sterile immunity unlikely. Chronic infection effects. Persistent malaria parasites in blood. Massively enlarged spleens, immune system impact. Hypergammaglobulinemia, polyclonal B cell activation, only fraction of antibodies specific for Plasmodium antigens. Immune response, contributors, regulatory T cells, cytotoxic T cells, GAN T cells, NK cells, monocytes. Cytokines, protective and pathological roles. Context for EBL emergence, uninterrupted malaria presence, continuous immune activation, splenomegaly, concurrent infections, viruses, bacteria, parasites. Key takeaways: malaria symptoms decrease with age in endemic areas. Immune system constantly activated by chronic malaria. EBL emerges due to persistent immune activation and concurrent infections. Summary: mean serum concentrations Fig 3a, total IgM concentration increases with age, peaks around 8 to 9 years, remains high. Fig 3b, total IgG concentration decreases initially, increases significantly after 2 to 3 years, peaks and remains high beyond 15 years. Population in malaria holoendemic area of Liberia. Key takeaways: IgM and IgG levels change with age in malaria endemic children. IgM peaks in late childhood. IgG initially decreases, then increases and peaks in adolescence. Summary: ubiquitous, potentially oncogenic gamma herpes virus infects 90% of adults worldwide. Infection characteristics: primary infection establishes lifelong persistent infection, low-level chronic lytic reactivation, viral shedding into saliva for transmission. Disease mechanism, usually benign, causes disease when host virus balance is upset. Depends on balance between viral latency and viral replication and host immune responses. Infection timing, African children infected early, seroconverted by 3 years of age. Affluent countries, primary infection often delayed until adolescence. Key takeaways: EBV is widespread and establishes lifelong infection. Disease occurs when host virus balance is disrupted. Early infection in Africa, delayed infection in affluent countries. Summary: EBV latency varies with differentiation stage, location, and origin of infected cell. Viral genome encodes DARS80 proteins, different sets of viral antigens expressed in each latency form. Latently infected B lymphocytes express 8 proteins and several non-translated RNAs, EBERs with anti-apoptotic effect. 6 nuclear antigens, EBNA1, 6, 2 membrane proteins, LMP1 and 2, 1. In vivo latency, established in resting memory B cells, infectious virus produced when memory B cells switch from latent to lytic phase. Memory B cells, no viral latent proteins, invisible to immune system. Tonsillar naive B cells, all 8 latent antigens, primary infection site after oral transmission. Epithelial cell tropism, EBV infected cells in oropharyngeal epithelium. In vitro, EBV infects any B cell type, transforming them into proliferative blasts. Key takeaways: EBV latency depends on cell type and stage. 8 proteins and non-translated RNAs involved in latency. Memory B cells are primary latency sites. Tonsillar naive B cells, primary infection sites. Epithelial cell tropism and broad B cell infectivity. Summary: latently infected B cells, virus can be induced to enter lytic cycle, inducing products, forbol esters, corticosteroids, anti-immunoglobulin. Key takeaways: various products can trigger EBV reactivation in latently infected B cells. Some 4A EBV DNA levels in plasma. Groups: acute malaria, AM, control Ghanaian children, CGCH, control Italian children, control Ghanaian adults. Acute malaria, high EBV DNA levels. Control groups, lower or undetectable EBV DNA levels. Percent EBV plus, AM 47%, CGCH 34%, CICHC 0%. Fig 4b, change in EBV load in Ugandan children with acute malaria. Measurement before days and after day 14, anti-malaria treatment. Saliva, EBV load decreases after treatment. Plasma, EBV load significantly decreases after treatment. Key takeaways: high EBV DNA in children with acute malaria. Anti-malaria treatment reduces EBV load in both saliva and plasma. Summary: lytic replication begins with the BZLF1 and BRLF1 expression, followed by translation of late genes, e.g., viral capsid antigen, VCA, reactivation monitoring, monitored by antibodies against VCA and BZLF1. Immune surveillance, dominated by CD8 plus MHC class 1 restricted cytotoxic T lymphocytes CTL, interferon IFN GVA responses to latent and lytic epitopes, primary infection response, directed against lytic antigens BZLF1 and BMLF1, EBV specific CD8 plus responses, secreting IL-10 immunosuppression, leads to increased viral load in blood, viral DNA detectable in plasma, not seen in healthy carriers. F1 initiate lytic replication, VCA and BZLF1 antibodies monitor reactivation, CD8 plus CTLs and IFN GAI play key roles in immune surveillance. Increased viral load in immunosuppressed individuals. Summary: Burkitt's lymphoma, BL chromosomal translocations, C-Mike on chromosome 8 with IG loci on chromosomes 12 or 22, molecular hallmark of BL. Mechanism: C-Mike, IG translocations, deregulation of C-Mike expression leads to abnormal activity of the proto-oncogene. C-Mice role linked to cell differentiation, proliferation, and apoptosis, not fully characterized. Detection in blood: C-Mike IG translocations in 10% of HIV-positive individuals, 2% of healthy controlled donors. Correlation: no established correlation between translocations and later lymphoma emergence. Key takeaways: C-Mike EG translocations are key in Burkitt's lymphoma. Deregulated C-Mike affects cell functions. Detected in some HIV-positive and healthy individuals. No clear link to later lymphoma development. Summary: malaria infections, polyclonal B-cell activation, hypergammaglobulinemia, production of autoantibodies, EBV and malaria, increased numbers of circulating EBV-carrying B cells in infected individuals. Study focus: impact of malaria infection on EBV persistence, interaction between P. falciparum and B cells where EBV persists. Key takeaways: malaria leads to B cell activation and increased EBV carrying B cells. Examines how malaria affects EBV persistence. Studies interaction between malaria parasite and B cells. Summary. P. falciparum malaria causes general immunosuppressive effects, EBV control. Acute malaria impairs immune control of EBV, leads to increased circulating EBV-carrying B cells. Mechanism. Result of increased proliferation of pre-existing infected B cells due to impaired T cell immunosurveillance and polyclonal activation, not primarily due to viral reactivation. Immunosuppressed patients. Viral replication contributes most to increased genome burden. Key takeaways. Acute malaria weakens immune control, increasing EBV-carrying B cells, mainly caused by B cell proliferation, not new viral reactivation. Immunosuppression increases viral replication. Summary. EBV DNA quantification, viral reactivation and increased viral burden common in acute malaria, high EBV DNA levels, detected in children, not adults, in malaria endemic areas, correlation between viral load and malaria endemicity. Study findings. EBV DNA in 31% of plasma, 79% of saliva samples from children with acute malaria, higher levels in infected children than in organ transplant or acute EBV patients. Comparison. Lowest EBV levels in control children, increased levels in acute malaria patients, very high levels in children with EBL. Anti-malaria treatment cleared plasma viral load in 85% of cases, did not affect saliva levels. HSV1 levels increased in saliva during malaria, reduced after treatment. Other human herpes viruses, HHV, in saliva unchanged. Key takeaways. Acute malaria linked to EBV reactivation and high viral burden. Children in endemic areas show high EBV DNA levels. Anti-malaria treatment reduces plasma but not saliva EBV load. HSV1 levels in saliva decrease after treatment. Summary. Mechanisms. Difficult to assess contributions to EBV persistence and increased EBL risk. T cell responses. Age-related loss of T cell IFN gang responses to EBV in Kenyan children in holoendemic areas. Acute malaria, augmented viral replication, increased viral load, presence of antibodies against BZLF1, viral reactivation, results from suppression of T cell and humoral responses. Possible direct activation by malarial antigens. EBV levels, high in serum and saliva during acute malaria, clearance of circulating EBV after anti-malaria treatment. Infection spread, produced virus may infect bystander B cells, increases infected B cell pool. Key takeaways. Acute malaria linked to EBV reactivation and high viral load. T cell suppression and malarial antigen interaction drive reactivation. Anti-malaria treatment reduces circulating EBV. Summary. Malaria impact on EBV, evident before full malaria immunity acquisition. Adults in endemic areas, no detectable EBV DNA in plasma. EBL characteristics. Childhood tumor. Peak incidence, ages 5 to 8. Malaria infection may impair virus-host balance at this age. Onset of EBL heralded by high antibody titers indicating viral replication. Key takeaways. Malaria affects EBV persistence before immunity develops. EBL peaks in children aged 5 to 8 with high viral activity indicated by antibodies. Summary. Malarial B-cell mitogens suggested by hypergammaglobulinemia, antibody production induced by IP falciparum-derived products. Polyclonal B-cell activation thought to involve T cells and accessory cells. Mechanism poorly understood. Study objective. Analyze impact of parasitized erythrocytes and purified malarial antigens on B cells from malaria-naive donors. Key takeaways. Malarial infection linked to B-cell activation, direct impact on B-cell homeostasis not well understood. Research focus, interaction between infected erythrocytes and B cells. Have our summary expressed during intererythrocytic proliferation of P. falciparum, presented on red blood cell RBC membrane. Binding to B cells binds to receptors on B cells, HIgG and HATIGM, HepHS, CD31 PCAM1, CD36, CIDR1A domain, PFE1 VAR1 from FCR3S1.2 clone. Multi-adhesive phenotype binds to CD31 PCAM1, CD36, and interaction details, CDR1A binds to surface immunoglobulins on B cells, binding dependent on PFEMP1 expression. No binding observed with FCR3S1.6 clone, low PFEMP1 levels. Recombinant CIDR1 binds to B cells, binding competed by soluble HIgM and HIG. Conclusion, physical interaction between infected erythrocytes and non-immune B cells. Mediated predominantly by CIDR1A domain of PAM1. Key takeaways, one crucial for RBC and B cell interaction. CIDR1A domain binds to multiple receptors on B cells. Interaction dependent on PFMP1 expression and involves surface immunoglobulins. Summary. Polyclonal B cell activator predominantly activates memory B cells. Binding specificity binds to non-immune HIgM and HIgG binds to IgG from human and other species. Affinity K values is KGM, HI 3.7 times 10C, and Memstis, HI 10 20 Memsflin, binds both FC and Fab parts of HIgM and AG. Characterization, identified as an immunoglobulin binding protein, IBP, similar binding pattern to S. aureus protein A. IBPs may divert the specific antibody response during infections. Key takeaways. PFEMP1 CIDR1 activates memory B cells and binds various immunoglobulins. High affinity for HIgM and HIgG functions similarly to bacterial IBPs like S. aureus protein A. Summary, i.e. CIDR1-Hofer interaction, induces B-cell activation and immunoglobulin secretion. Experimental findings, IE and recombinant CIDR1 induced B-cell proliferation and activation in co-incubation assays. Pre-incubation with soluble HIGM inhibited cell proliferation dose-dependently. Activation indicators, increase in cell size, upregulation of HLA-DR, CD33, CD40, 54, CD58 Tate, CD80, CD86, production of soluble IgM, IL-6, IL-10 in vivo context, PFEMP1, Na, present in bloodstream and immune system, expressed on infected erythrocytes, importance of antigen affinity, avidity, and density in B-cell activation. Affinity classification, CIDR1 Na classified as low affinity in antigen, Ka 1 times 10 to the megamer n, T-cell independent responses. Large differences in affinity have small variations in B-cell response. Key takeaways, AHA activates B cells and induces immunoglobulin secretion. Pre-incubation with HIGM reduces proliferation. Low affinity antigen, but still significant in B-cell activation. Summary, anchored on infected erythrocytes in knobs. Knobs, electron-dense structures, knobby phenotype, common in most P. falciparum clinical isolates. In vitro adapted parasites, e.g. FCR3S1.2, may lose knobby phenotype due to gene deletions on chromosome 2. Function of knobs, augment PMP1 adhesion under flow, increase surface protein density, B-cell interaction. Knobby infected erythrocytes create microenvironment, high amounts of PFEMP1, CL1 alpha near BCR clusters. Key takeaways, knobs enhance PMP1 adhesion on infected erythrocytes. In vivo interactions with B cells involve knobby erythrocytes and PFEMP1 proximity to BCR clusters. Summary. CDAR1-induced activation, B-cell activation and proliferation without accessory cells or cytokines. Polyclonal activation in vivo may be enhanced by other cofactors. Interaction sites. P. falciparum infected erythrocytes interact with B cells in circulation and other compartments. Mature infected erythrocytes sequester in a vasculature, avoid spleen clearance. Under flow conditions, many infected erythrocytes trapped in spleen. Splenic environment, B lymphocytes can be up to 50% of splenocytes. Splenic enlargement due to phagocytic cell expansion favors interactions between B cells and infected erythrocytes. Phagocytosis and antigen presentation. Infected erythrocytes phagocytosed, antigens presented in soluble forms or as immunocomplexes by dendritic cells, presence of cytokines, just T helper cells, and co-stimulatory signals enhances activation. Molecular mechanisms. PFMP1 CIDR1 induces polyclonal activation, leads to hypergammaglobulinemia, results in deflected immune response to malarial antigens. Key takeaways. Activates B cells without accessory factors. Splenic interactions crucial for polyclonal activation. Leads to hypergammaglobulinemia and altered immune response during malaria infection. Summary: CDR1 as polyclonal activator, tested on naive IgD plus CD27- DAS and memory CD27 plus B cell populations. Memory B cells activation, increased proportion expressing CD70 and CD95, upregulation of CD70, CD95, CD69, CD86. Naive B cells, no effect on expression levels of the same markers. Acute malaria infection, increased memory B cells subset, decreased naive B cells subset compared to post-recovery. Preferential activation, CIDR1A domain of FM1 preferentially activates memory B cells. Key takeaways: CIDR1 preferentially activates memory B cells. No change in naive B cells marker expression. Acute malaria increases memory B cells, decreases naive B cells. Summary: CR1A and germinal center B cells. Affects dynamics of GCC B cells in secondary lymphoid tissue tonsils. Reduces apoptosis G0 G1 phase. Increases proportion of cycling cells S plus G2M phase. Gene expression, upregulation of genes in immunological signaling pathways, cell growth, apoptosis. Transcription, anti-apoptotic genes, upregulated CALB1, HSP40, GSTM2, protein level changes, downregulated BCLXL, BCL2, increased XIAP. Mechanism, increased B cell survival mediated by interference with BCLXL, BCL2 anti-apoptotic pathway, inducing expression of XIAP. Key takeaways: CDR1 enhances GCB cell survival by reducing apoptosis and increasing cell cycling, upregulates genes involved in growth and apoptosis regulation, increases expression of anti-apoptotic regulators. Summary: B cell receptor, BCR, complex of membrane immunoglobulins, heavy and light chains, associated with IgD beta heterodimers, CD79A, CD79B. Function: signaling subunits couple receptor to intracellular signal transducers. Oligomerization triggers signal cascades leading to cellular response. T cell independent activation, initiated by binding of multivalent antigens to BCR. CIDR1 activation, soluble CIDR1 activates B cells and induces proliferation, process partially mediated by surface immunoglobulins. Additional molecules. Binding not fully competed by soluble immunoglobulins suggests involvement of other molecules, supported by different gene expression profiles between CIDR1 FON and anti-human immunoglobulin stimuli. Key takeaways: BCR involves IgA for signaling. CR1A activates B cells through BCR, possibly involving additional molecules. Gene expression differences indicate unique activation mechanisms. Summary: associated with naive B cell phenotype, expression lost as B cells differentiate into memory cells, acts as a co-receptor that negatively regulates BCR signaling. CIDR1 Fi1 binding binds to CD31, hypothesis, Preferential activation of memory B cells because on naive B cells, CIDR1A binds both BCR and CD31, antagonizing BCR activation. Implications. Important for understanding malaria's role in EBV persistence and EBL pathogenesis. EBV persists and establishes latency in the memory B cell compartment. Key takeaways. CD31 inhibits BCR signaling in naive B cells. CIDR1A preferentially activates memory B cells by avoiding CD31's negative regulation. Critical for understanding EBV latency in the context of malaria and EBL. Summary. CDR1 FAF of PM1 reactivates EBV in memory B cells, direct interaction with B lymphocytes and IE or CIDR1 HAFI. Mechanism. CIDR1 mediated activation involves surface immunoglobulins, hypothesized to trigger viral reactivation similar to anti-human immunoglobulin. Experiment. BL cell line Akata used as model, CIDR1A bound to Akata cells, increased viral load in CIDR1A stimulated Akata cells, real-time PCR. Results. Increased viral load, not due to increased EBV carrying cell numbers. Co-incubation with CIDR1A and pan-caspase inhibitors VOD showed similar EBV production. Key takeaways. CIDR1-Ta reactivates EBV in memory B cells. Activation involves surface immunoglobulins. BL cell line Akata confirms increased viral load with CIDR1A stimulation. Summary. Figure 5 analysis. P. falciparum infected erythrocytes and CI1HA of PFE1 bind to B cells. Images A, B, C. A. Ethidium bromide stained P. falciparum PRBC, red-yellow, with acridine orange stained B cells, green. B. FCR3S1.2 clone with B cells. C. Uninfected erythrocytes with B cells. Fluorescence activated cell sorting D. Medium control, black. Medium with recombinant GST, red. GST-DBL1, green. GSCIDR1, blue. Key takeaways. Infected erythrocytes and CIDR1 hub bind to B cells. Fluorescence analysis differentiates binding interactions. Figure 5 visually and quantitatively demonstrates binding specificity. Summary. Figure 6 analysis. P. falciparum PRBCs and ZR1-SA bind to EBV positive B cell line Akata. Images A, B, C. A. Akata cells, green, with RBC stained with PK26, red. B, C. Akata cells with purified PRBC. Binding evaluated by light microscopy and immunofluorescence microscopy. C. Magnification of binding, white arrows using phase contrast microscopy. Binding evaluation, DEF. Akata cells incubated with soluble GST-D or CDR1-HE, stained with anti-GST AB and anti-mouse Alexa 488 conjugated AB, evaluated by light microscopy, left, and immunofluorescence microscopy, right. Facts analysis, key takeaways. CDR1-HA binds to EBV positive B cells. Akata, immunofluorescence and light microscopy confirm binding. Facts analysis shows binding specificity and interaction strength. Summary: lytic cycle reactivation. Demonstrated using a kata-line-based system. GFP expression indicates lytic cycle induction. CIDR1A stimulation increases GFP positive cells compared to controls. EBV load correlates with proportion of lytic replication cells. Viral reactivation induced in latently infected B cells from healthy EBV carriers and EBL patients. Conclusion: CIDR1.MI domain induces EBV reactivation in a kata model. Key takeaways: CR1HAR induces EBV lytic cycle reactivation, GFP expression used as indicator, effective in both healthy carriers and EBL patients. Summary: first demonstration, malarial antigens, CIDR1A of PFE1, drive latently infected B cells into EBV replication. EBV reactivation is modest but consistent. Proposed mechanism: CIDR1HA interactions with surface immunoglobulins mimic BCR cross-linking, may involve other molecules. Primary interactions occur in secondary lymphoid tissues, e.g. spleen. Memory B cells potentially latently infected by EBV undergo maturation. Malaria infection disrupts secondary lymphoid organs, favors viral reactivation in EBV-carrying lymphocytes. Key takeaways: CIDR1HAR can induce EBV replication in latently infected B cells. Mechanism involves surface immunoglobulins and BCR mimicry. Interaction likely occurs in spleen during malaria infection. Summary: Figure 7 analysis, stimulation of tonsil B cells and PBMCs from EBV-positive donors and BL patients. Conditions: medium alone white bar, GST, light gray bar, CDR1 dark gray bar. Results: CIDR1 significantly increases EBV load. EBV genomes measured by real-time PCR after 48 hours. Key takeaways: CDR1 increases EBV load in B cells from EBV-positive donors and BL patients, measured using real-time PCR after 48 hours incubation. Stimulation with cyanogen shows higher EBV genome copies compared to medium and GST. Summary: malaria and EBV are closely linked. Endemic Burkitt's lymphoma, EBL, emerges from malaria parasites, immune activation, splenomegaly, EBV infection. Augmented EBV load results from impaired EBV-specific T-cell response, polyclonal B-cell activation, viral reactivation driven by malarial antigens. Possible mechanisms: differentiation of memory B cells where EBV persists. CID1 induces B-cell activation and immunoglobulin secretion. IG binding capacity of PM1CR1 could mimic anti-IG and rheumatoid factor, leading to viral replication. Key takeaways: malaria significantly impacts EBV reactivation and EBL emergence. EBV reactivation mechanisms involve memory B cell differentiation and IG binding by malarial antigens. Summary: viral loads, higher than those in primary EBV infection or immunosuppressed patients. Increased viral load is a risk factor for EBL development. High risk, children infected early in life with high antibody titers to virolytic antigens. Augmented virus production contributes to EBL lymphomagenesis. Mechanisms: 1. Infection of bystander B cells, increased infected B cell pool, GC reactions. 2. Increased survival of infected cells due to anti-apoptotic products, EBNA1 viral RNAs, downregulation of pro-apoptotic protein BIM by EBNA3A and 3C. Key takeaways: higher viral loads linked to EBL development. Early infection with high antibody titers increases risk. Mechanisms include infection of bystander B cells and increased cell survival. Summary: high levels, circulating viral EBV DNA and malaria parasites. Implications suggests long-term EBV reactivation in children in malaria endemic areas. Represents a different pattern of viral persistence compared to high-income countries. Key takeaways: high EBV DNA and malaria parasites coexist in malaria endemic areas. Indicates long-term EBV reactivation, distinct from patterns in high-income countries. Summary: BL pathogenesis key event, activation of c-MYC growth promotion, inhibition of c-MYC-induced apoptosis. CEDR1 role, rescues B cells from apoptosis, promotes cell cycle entry. Deregulated c-MYC, decreased apoptotic sensitivity, more cells tolerate increased MYC translocations. Genetic findings: small percentage of healthy individuals carry c-MYC-Ig translocation without lymphoma risk. Indicates c-MYC rearrangement alone insufficient for malignancy. Multiple genetic lesions needed for C translocated cell survival and immortality. Lymphoma pathogenesis: prolonged lifespan of translocated cells, genetic instability from MyC overexpression, active proliferation increases genetic lesion occurrence. Key takeaways: c-MyC activation critical in BL pathogenesis. CIDR1 aids in B cell survival and cell cycle entry. c-MyC Ig translocation alone not sufficient for malignancy, requires multiple genetic lesions. Summary: polyclonal activator, CDR1 in malaria parasite, malaria infection in holoendemic areas, repeated protracted episodes, chronic B cell stimulation increases mutation possibilities, aid enzyme, activation induced deaminase involved in somatic hypermutation, and isotype switching. EBV role, EBV turns on AID. Key takeaways: one line on chronic malaria infections stimulate B cells, increased mutations via AID enzyme, EBV activates AID. Summary: model proposal. Malaria contributes to increased EBL risk. Mechanisms: uninterrupted presence of parasites and antigens, splenomegaly, trapping accumulation of antigens in secondary lymphoid organs, e.g. spleen, hyperactivation of germinal centers, increased somatic recombination, c-Myc Ig chromosomal rearrangements. CIDR1 affects proliferation of B cells carrying translocations, additional genetic lesions, prolonged lifespan of B cells with translocations, increased B cell infection by EBV. Cytokine environment, IL-10, IL-4, IL-6 promote cell survival, incites polyclonal B cell activation and EBV reactivation in malaria, pathogen cooperation in lymphoma pathogenesis. Malaria promotes EBL risk through germinal center hyperactivation and c-Myc Ig rearrangements. CR1 induces B cell proliferation and translocations. Favorable cytokine environment enhances B cell survival and EBV reactivation. Summary, model explanation, malaria infections, holoendemic areas, parasites and soluble antigens in bloodstream, splenomegaly, accumulation of antigens in lymphoid organs, e.g. spleen, germinal center hyperactivation. Mechanisms, increased somatic recombination, c-Myc Ig rearrangements, CIDR1A induced proliferation, B cells with translocations, additional genetic lesions, lifespan prolongation, protection from apoptosis, increased EBV infection frequency, favorable cytokine environment, IL-10, IL-4, IL-6. Splenomegaly and germinal center hyperactivation due to malaria, CIDR1A increases B cell proliferation and translocations, enhanced survival and EBV reactivation facilitated by cytokine environment.